We are about to bring on the single greatest science communicator on Twitch. His name is Professor Danny Anduza. He is the reason we're here on Twitch. He's an inspiration to everyone who should be is a scientist. The man knows the true meaning of being a scientist. He spreads his love of science here on Twitch, does daily science content, talking dinosaurs. If you love dinosaurs, if you love science, go follow Danny. If you're not sure, you're going to be sure in about 2 shakes of a leg when we bring him on. And you immediately will be like, this guy is the absolute best. So yeah, welcome the heck in, everybody. Boji, I have no idea, but welcome. Hello. Guys, I'm going to go ahead and invite Danny now. Got the virtual camera set up. Let's see. Horizontal. Let's send that invite to Danny. And you did get the sticker. Nice, Danny. Y'all, I was just like, oh, I think Danny might like these stickers. Let me send him a sticker. In my defense, y'all, I sent one to Lordy and iOS too, okay? It's totally fine. Alright, let's see if we can get Danny on with some audio as well. Hi, Danny.
Hey there, Blint. How you doing? Can you hear me?
I can. Thank you, sir. How are you doing?
I'm doing really well. Thanks. Yeah. Happy World Frog Day to you.
You too. Do you have a favorite frog?
Oh, boy. I'm partial to the gliding frogs of Borneo.
I saw y'all were watching a video on them, right? The slow motion flying through.
I think they are just so cool. Gliding behavior in animals I just find really, really fascinating, and so it's so cool that those critters exist, you know?
And they're beautiful, too, and the footage...
They really are, yeah.
Like, full disclosure, Danny, I may have had you up to the computer next to me, as I always do while we're live.
I was wondering how you can be live and also type in my chat at the same time.
I can't.
I don't know how you do it. Yeah.
It's a lot of. It's just because I love what you do and I want to be like whatever I can to support. You know.
Well, I really appreciate that, Blint. You make me blush, and again, I'm in awe of your ability to multitask and get things done. And 3 jobs, streaming full time, caring for a child, you know, I don't know how you do it. It's amazing. So
I appreciate you, my friend.
I'm in awe.
And for those of y'all saying the audio might be a little bit low on Danny, we have it maxed out on his side, or at least like what I can do on my end. He's at 200%. So y'all let me know. I did crank the desktop audio a little bit, and maybe that feed is a little bit better. But y'all just...
My microphone is about as high as it can go right now and still be reasonable, so I'm not peaking or anything.
Yeah.
Yeah.
Let us know, y'all.
Talk close to the mic.
It's some Danny ASMR.
Yeah, there we go.
So y'all just let me know, y'all, how the audio is, and sometimes it just cuts in and out because it's guest star, and then it ends up being okay.
It could be a little buggy at times, but it usually kind of repairs itself.
Yeah,
I don't know how it works. It's like a living organism, you know. It's regenerating.
Oh, Sparky says she can hear you fine, so I think we're set.
All right.
Yeah, it's my favorite is when we have the echo and neither of us touch anything. And then it just fixes itself.
Yep.
But y'all, we have... So, Danny and I, for those of you who are new here, we exchange papers during the week of our respective fields, read them, and then we just talk about them together and take your questions. And what's really cool is, you know, I'm a geneticist. I don't know anything about fossils beyond what Danny has taught us on his stream, right?
And I don't know anything about genetics. I mean, shoot, I am in the dark about that. So it's really cool to be able to have this exchange like that.
Yeah, so it's like asking the questions like from our unique perspectives that maybe no one else would really ask, because why would you ask that kind of question unless you're in your respective field? And it's like, like some things, Danny will say like, of course that makes sense. Like why is this experiment necessarily revolutionary? Because yeah, rats can have imagination, sure. And it's like the different thresholds for experiments and how we take them, and so it's my favorite part of the week. But this week, Danny,
really a highlight for me too. Yeah. Oh man, I'm excited for this crossover, Valent. This is there's a nice confluence of different subjects and things that are directly related to your field. Talking about the evolution of angiosperm plants and then how that influenced ant evolution, and this is cool. We're gonna have more common ground than usual, I think, in today's stream.
But you know what's wild to me is neither of us mentioned it to each other. Right? We just so happened to have sent each other ancient plant themed things.
Well, I think didn't we talk briefly about the angiosperm paper last week? I said we should cover that next week or something.
No, well,
I remember thinking. I don't know if I said it out loud. But
I don't think so.
Okay. Yeah.
But either way, I had not remembered it if we hadn't talked about it. So it independently came to the. I was like, oh, I think this would be a fun paper, and
it's going to be beautiful. Oh man, I can't wait, because the cycad paper also ties into that too, because the rise of angiosperms seems to coincide with the decline of the gymnosperms, including the cycads. So
that's why when I saw it, I'm like, that's a great link. And for those y'all are interested, we will post the papers into the chat. However, because I'm a party pooper, the paper I picked is behind a paywall. But on the Discord,
it's not your fault.
It shouldn't be, right? On the Discord, y'all. Willie, thank you for the follow. On the Discord, I did post the PDF. So if anyone does want the paper, the PDF is there for download. Just don't tell the PNAS folks that we did that.
Yeah.
I went 4 different routes trying to get that paper today, with emailing someone who has the access to it, and apparently their university didn't have it yet. I got another university friend, and they were like, it hasn't been pre-released, even though they were subscribed to PNAS. I ended up getting it from the authors. So that's one thing.
Very nice.
Yeah.
That's cool.
I think, and you've talked about it on your stream, right, Danny, that the opportunity to go and get papers by just emailing the person who did the research.
That is, I think, a lovely piece of advice. If anybody watching right now, if you ever hear there's a news article about a cool scientific discovery, it's a new paper, and you go and you click on the paper and it's paywalled, just see if you can find the email address for the authors and just shoot them a quick email and say, hey, I'm really interested in reading your paper. Would you mind sending me a PDF? And you'd be surprised how often they reply and they say, of course, here you go. Enjoy. Because we as scientists, it's not like we're profiting off of the paywall or anything like that. We want people to read our work. We're doing the work for free, giving it to the journal for free, and we want people to appreciate it and enjoy it and read it. So,
and
email the scientists.
I don't know if you've had this, Danny, but people relatives asking if I rent the article or pay for it, do you get that money? I don't know if you've had that exchange too, but
not with family members, because I don't know. They don't really know what scientific journals are, but with members of chat, for sure. It's like, yeah, I found your Trier Archuncus paper in the Journal of Cretaceous Research. How much of that money do you get if I pay $79.99 to rent it for 36 hours or whatever? It's like, they didn't even send me a copy of the paper. Yeah, let alone getting paid for it. I don't see any of that money.
It's one of those things too that's not really talked about when it comes to science communication. And the wild thing to me is that it is funded by taxpayer money, and that it's not then available to the taxpayer, which is really frustrating, y'all. But then your workaround is you can go in and request it from the authors. That's what I did. I clicked on Vincent's name here, and it popped his email address, and I shot him an email. I'd be like,
nice.
I would love to have you read your paper. And so he's like, oh yeah, sure, here's the paper.
Nice.
Yeah.
Did you specify that you're also a researcher, or did you
I did mention like I'm an ant geneticist, and I thought,
Which
I
you
was
don't
like,
have to. I kind of feel like, as a member of the general public, you might be more likely to get a response if you're not a researcher. It's just like, hey, I saw the press on this and it looks really cool. I'm just, a person. Because I've before had researchers on rare occasions email me back and say, hey, is there any way you can try and get it through an institution or something? Like, if you were a member of the general public, I'd just send it to you right away. But I don't know, but like, yeah, it's been interesting, some of the responses that I've gotten. But like 95% of the time, they just send it to you right away if they have it. Although this week, Belin, or rather last week, I emailed some researchers to ask for a copy of their paper, and they said, we'd love to give it to you, but unfortunately, the journal didn't give us a copy either. So they didn't have a PDF. So instead, they sent me like a draft manuscript, which was really nice. But yeah,
really?
yeah, uh-huh.
That's
Yep.
so strange
Yep.
to me.
It happens. And it's in a journal that I've published in before, actually. And they're kind of notorious for this. But yeah.
That's a big yikes. Because you need, right? You should get like proof so that you can check everything, and then you get the final version in theory, so you can make sure everything is appropriate. Because y'all, some silly things like the figures can be inappropriately placed, or like lines will shift around because of the file format, and you can have a figure that's inappropriately labeled, not because of the researchers, but just because during the copy edit process.
The formatting, yeah.
Yeah. So that's oof. Well.
So I'm waiting on the. We'll go over that paper once they send me the finished copy. Once they manage to get hold of it. The authors. Oh boy.
It's.
Yeah.
Why is that a thing, Danny?
It's, I don't know. I can talk about that all day. I'm resisting the urge.
Shall we start with your plant paper?
I was kind of thinking we start with your ant paper.
Okay.
Your ant and plant paper.
Okay.
If that's good with you.
Yeah,
Oh,
of
and
course.
I've got just a little bit over 2 hours that I can spend before I gotta go eat dinner.
Okay, so we're around 11, we'll say?
Yeah.
11 my time?
11:15 your time, yeah.
Okay,
Yeah.
cool. I just want to, double, triple check.
Yeah. I appreciate that.
Okay, oh, no,
Yeah.
sir, thank you. So yeah, here's... I like this... Caught my eye for a couple of different reasons, Chad. Number one, it's ants.
Uh-huh.
Number two, it has the hell ant in there, which is one of my favorite species. And then I think it's really well written in that it presents a couple of different hypotheses, tests them, and even counter-argues existing hypotheses and why they might be wrong, using an absurd amount of evidence. Like, they looked at a ton of different fossilized remains, 24,000 fossilized pieces of evidence, 14,000 modern-day occurrences of ants.
That is a huge sample size for paleontology, Blend. I hope you realize that, that might be impressive from your perspective as a geneticist, but as a paleontologist, that is mind-blowing.
So that's what I wanted to ask you to start
Yeah.
with, is like, I have never seen something like this in terms of just the numbers.
Yeah. I was actually surprised that this paper didn't have 40 authors on it.
Oh yeah,
Normally, when you've got something that's that comprehensive, you would have a huge author list. In here they've got what, 4?
Yeah.
4 or 5? Yeah, it's
and it's
surprising.
It's not even that their acknowledgments are especially insane, with 300 undergrads, right? And it's not they're not giving credit for people's work. It just is someone shared their data for us. They gave us institutional collections that they took from.
Yep.
And then, private collections that they looked at.
Mm-hmm.
So it seems like they analyzed all of it.
I got a really strong sense that this was a real passion project for somebody. One or two of the authors in here, they probably just eat, live, and breathe fossil ants. And this is their chance to really make a big difference in the field. My guess is the timeline for this paper probably goes back a decade or more.
I would guess they've probably been working on this for a long time.
It's just the God, the numbers alone are kind of mind blowing to me. I wish, like, I don't know if you looked at the supplement too, but they have like a breakdown of all the different types of individuals that they had as well. And the only thing I would have loved to have is images. Honestly, like, that's the one thing that I'm like, there's not really any pretty pictures of any of the samples or like what representative samples might look like.
Shoot, yeah. I wonder if that's a journal limitation too, if they like just
So-
don't allow for that.
They have, let's see, it was like 38 supplementary figures. So, and like I've published in PNAS before, and it's like these are all the figures, like these are these different graphs and showing like different types of ant evolution over time, and there's never been I don't think they actually have a limit on the number of figures like that can go into supplement or type. Maybe it was just it got so unruly that they were like, we don't want like 800 supplementary figures.
That would make sense. Yeah, shoot. And if that's another barrier to publication too, like it would just take too long to put that together, then I understand that.
Yeah.
I wonder if maybe they're working on a giant monograph or something like that that would have all the figures included, because that would be really cool.
Or like a poster even. Like just a cool poster to convey the images, and like, because if you already took them and collected them and categorized them for 24,000 fossils, and a lot of them
they already have the images all labeled and everything. Like presumably that already exists, and they just have to format it a little bit. So,
and a lot of these were in amber too, which was really cool. That chat, like on Mondays we look at insects in amber, and we've looked at ants, and so it's kind of wild to think that they also were using similar data to what we are doing live on Twitch on Mondays, like, because it's unknown samples we look at, and that's kind of what they were doing too. It's like you said, Danny, there's no, in the press release either, there wasn't like a sense of time of how long something like this took, but I would love to have heard like and science streams. Welcome the heck in, Raiders. I hope y'all had a lovely stream. I saw y'all were talking about population effects today. I hope y'all had a great one. Tell us how it went. Go check out y'all our namesake of also science streams. Welcome on in, y'all. For the Raiders coming in, my name is Balint. This is the lovely Professor Danny Anduza of Paleontologizing. You can go follow his name. It's in the hot link description today. We're having a crossover talking fossil science and genetics, and we're talking about this really cool paper that just came out, I think this week, on how the Angiosperm Terrestrial Revolution, or ATR, saved ants from extinction. Is basically the end conclusion of this. And we'll be talking about, correct me if I'm wrong, Danny, the idea of a living fossil not being real.
Basically, at least not for cycads. It's going to be a fun one, I think, to discuss. So, and it ties in with the Angiosperm Revolution paper as well. So
I'm excited for it. Science stream that we had a good review of Hardy Weinberg Equilibrium. Nice, very very cool. Well, thank you so much for the raid, and welcome on in, Raiders. We're just getting started on this paper and just going over the numbers that the researchers looked at: 24,000 fossilized ants and 14,000 modern living ants, and they were actually testing a hypothesis back from 2006. And actually, I know, Danny, you were interested in this study. I sent you the original paper too that they were referring. Corey Moreau, that's a name that I knew. So this is, let's see if I have her paper pulled up too. Here it is: the phylogeny of ants, diversification in the age of angiosperms. Was the original paper back in 2006. And Corey, now, it was when she was a graduate student. She's now a head of a lab at Cornell. Know a lot for turtlehead ants. And they put forth this hypothesis of the reason that we have so many different ant species today. Just to give y'all an estimate, there's 16,000 species of ants alive today, with an estimated 16,000 that have not been discovered.
That's impressive.
Right. And so it's like, well, where did all these animals come from? Like, why are there so many? And so this paper was trying to link that diversification and that mass of ants from the rise of the angiosperm. So those are the flowering, fruiting-bodied plants that, have fruits. It is anonymous. Oh my goodness, we love this stream. Thank you very much, Anonymous. I appreciate you. That's the second one of those gifts that we got in 2 days. It's crazy. Appreciate you. Hi, Nelf. Welcome in.
Nice.
So, with that many different ant species, like, they weren't doing that kind of as big of a study as was done here. This one, like the previous study, I believe they had 43 fossils.
Which is good. Like, that would actually be a pretty good sample size for a paleontological study. But holy cow, that's nothing compared to this current one.
Yeah, and so it's because I looked back at the original paper and I was like, that doesn't seem like it's too bad, given like what we've talked about, where
yep,
right, a dinosaur can be named off of a bone fragment.
Occasionally, yeah. It's not best practice, but it happens a lot. Yeah.
And so it's just like, 43 seems pretty good, but no, let's up the game to 20,000+ samples.
Holy cow. Yeah, yeah.
And so, like, that was, I think, just putting them into perspective, right? Like, having that, the perspective of this is what is being built upon, and this was considered not really enough data to come to a strong conclusion. I think it's an interesting perspective to have from the side of the researchers, right?
Yeah.
And so they wanted to have different models of how the ants may have come to be. Like, they knew, okay, there's a ton of ants, but how do we have them? And they presented these 4 hypotheses, and I'll zoom in, y'all, so we can see. But they also had a fifth one, right? That was they didn't even bother graphing because they were just like, this totally is disproven by the data. So which makes sense. It was they had we can go into the null hypothesis first, and that was that there was resource competition between what are called crown ants and stem ants. So stem ants, let me get y'all actually a picture. Open image in a new tab. Here we go. This is one of my favorite ants, y'all. This is the hell ant. And it has these vertical mandibles. Oh my goodness, 6-foot ant is in the chat. Yes, you are on screen, 6-foot ant.
Nice.
If ever there be a place that you end up that is serendipitous, welcome on in. But so, I don't know, Danny, if you've seen the sample of the hell ant that we have, but it has these vertical mandibles, and that's a sign of what we think is hyper-specialization.
Right, yeah.
And so one of the hypotheses as to why this animal went extinct and the crown ants or your modern ants survived is because they just were competing for the same resources. And so that actually has been debunked now with the rise of we'll look at the data, but they'll show too that with the rise of the angiosperms plants, like these hell ants or stem ants as they're referred to in this paper, plus your modern day ants were still coexisting at the same time, and they fed on and had very different diets.
Yeah,
and so
that makes a lot of sense. And yeah. I might want to explain for the chat too, just the concept of like stem taxa. Yeah, so this is a thing in evolutionary biology. You've got, like when a new group evolves, you've got kind of the ancestors at the bottom that are the stem group. So it's like the stem of a tree, the stem of a plant. And then you've got the crown group. So like a stem mammal, for instance, would be like Dimetrodon. That's like a stem mammal. It's like a proto-mammal. It's like, one of those critters that's closely related to the ancestors of the mammals. So like Dimetrodon or Edaphosaurus would be stem mammals. And then you got the crown group mammals, which are like, that's the common ancestor of all of your modern mammals.
plus all of its descendants. That's the crown group, mammals. Think of it kind of like, I guess you could think of Velociraptor as being like a stem bird, because it's closely related to the ancestors of birds. Because the group that gave rise to birds, the group of dinosaurs, probably the Troodontids or something like that. Might be the common ancestor of Troodontids and Dromaeosaurs. Anyway, when you think stem, think like primitive, or like these are the ones that were kind of weird that were there a long time ago. They're not direct ancestors to the modern group, but they were hanging around back then and doing weird, cool stuff.
And here's the phylogenetic tree to illustrate that same thing. So it actually even says, so this is a very different paper that kind of informed the hunting behavior of your hell ants. But here it's that ancestral state right there of the hell ant that we were looking at. And then your crown ants, which are your modern like carpenter ants, are all in this pink zone here. And yeah, it's the same nomenclature too, y'all. Like it's cool to see like how you just explained it, Danny, and then it's like cross about the different papers, like referring to it in the right way, which is really cool.
Yeah. So I just wanted to clarify that for anybody who is curious.
No, absolutely. Thank you, Danny. Sloppy says I like that. The authors mention also about the idea that the age of lineage increases its likelihood of going extinct. Oh, gotcha. Nice. Very, very cool. Yeah, it's sloppy. They, I think, have a lot of good counter explanations in this paper, which I thought was neat. And then I'll highlight for y'all right here. This paper came out. We looked at the precursor paper from Dr. Moreau's lab, where they didn't even have hell ants really yet in terms of sampling. There had been like a sample, but this was the first one in 2019 where you got to see the hunting behavior, and this is where it was found out that these vertical mandibles were hyper specialized for hunting, and then the prey started to go extinct, and then sure enough the ants went extinct too. So it's not that they were co-competing with your modern day angiosperm related ants. It was their prey that was very different and niche specific went extinct.
I wouldn't be shocked if like the prey of the hell ants were like other insects that were specialized on certain kinds of gymnosperm plant, where like they're heavily reliant upon a certain species of cycad or ginkgo or something like that, and then when those groups take a tumble, hell ants do too, because shoot, what are they going to eat?
Unfortunately, I don't think we have that resolution of data to know like what these
grub,
it's a really
difficult one to test.
Grub beetle-like creatures ate, but I really like that hypothesis because that would be.
It's a nice just so story there, but it's a tough one to test. Yeah.
And Sparky's wondering if that's moon pie.
This is moon pie here, yes indeed. Yeah, we're a little white spot right there. Yeah, thanks for visiting, moon pie. Don't drink my drink. Get out of it. Stop it.
See, Noodle drinks my drink, but she dips her paw into it. She doesn't drink it like a regular cat, and so she'll sample. But then, if Lita leaves her coffee out, she puts her paw in, and if there's any cat litter between her toes, right, no. you'll go to the coffee, and you'll see these little things floating. And you're like, well, that's it for that coffee.
Oh, jeez. That's really. I've never really seen a cat do that though in person. I've seen it in videos.
We haven't either. Yeah. She's the first one that does that, and she just everything. Funny. She'll just it's very classy looking, but
yeah.
6-foot ants. Wonder maybe I missed this, but I'm so baffled. How do you get vertical mandibles from normal ones? Oh, that's so 6 foot.
Ooh.
If we look back at this graph, it shows you the evolution of these mandibles, and that orange region in each one of these images is the mandible. And so the ancestral state, like this very early lineage, they develop these vertical mandibles, and you can see there's a variety of them. So there's ones that are pointed a little bit upward, and then others that are much more vertical, and those are just specialized on a particular kind of prey. and then that orange region further evolved to what our modern day mandibles look like with horizontal opening. So it's not that it came from a different limb, it's that same structure. It's just evolved for a specialized purpose. So I think it's really cool to see when an animal is evolving for niche something. So like there's a kind of environmental cue and factor that allows them to do really well in a particular scenario. And so these vertical that's why they're called the hell ant too, 6 foot, because they just look terrifying. And they caught things with those vertical mandibles, but we think they went extinct because the prey died out. And those vertical mandibles are so specialized that what are you going to catch? Like, you can't de-evolve almost. Like, I don't really like that phrase, but that's basically what you'd have to be doing to be able to have those horizontal mandibles in a time frame that actually allows them to survive. And that just wouldn't be... Yeah, they were over-specialized, exactly, Hanny. Too specialized for it.
What I'm wondering about, Belent, is when you're looking at modern ants, are there any species of modern ant that have hypertrophied mandibles, really, really big mandibles, that are associated with predation?
So one here that on the graph, this L. See if I can blow it up a little bit. There are some of them, and they tend to have also this more wasp-like body, like the hell ant. So, the evolutionary origin of wasp, or for ants and bees, stem from wasps, and so seeing this wasp-like body that's very pointed on the abdomen with like a stinger is more of this like ancestral state, we think. And so there are some like this one here, the bull ant, the jumping ants, that have these really, really long mandibles. They're not curved, they still open horizontally, but they're really long. And the other thing too is we think the hell ant, you can't tell the difference between male or between queen and worker. That's based on the little fossil evidence that we have, that there's never an instance where if you find a queen, the only way you can tell is that there's little wing nubs on her. But physically they look the same, and that's something that we see in our modern day ants as well with these, where workers and queens look identical and they can actually switch castes depending on need of the colony, which is not true for your carpenter ants or more specialized looking ones.
You don't have any reason to suspect that hell ants were not eusocial, do you?
So oh god, you caught me, Danny. You caught me.
Sorry, I'm not trying to catch
No. This is a really good point, and this is, I think, similar to how, like, whenever I also ask you these like kind of questions of like, well, how many samples do you have? It's the So if we're using the Harbignathus jumping ant, which looks similar in terms of long mandibles, as a proxy for behavior, those ants don't live in high densities now in modern times, and so the likelihood of catching these modern ants in amber with multiple individuals is insanely low.
I see what you mean. Yeah.
And
it's
so
like only when you have the only reason you have a crowd of ants getting caught in amber is if they're eusocial, if they're hanging out in big groups anyway.
Yeah, and if they're already living in a large group. So it's like these Indian jumping ants, it's not that they're not eusocial, it's just that their numbers are fundamentally lower than if a carpenter ant colony got caught. So that's point one. Point two, again, using modern day ants as a proxy, which may or may not be appropriate. These ants that look very similar and behave what we suspect similarly live underground. And so it's just hard for tree resin to run into underground tunnels.
Yeah, of course.
So that's another point of it. And then, the fossil evidence right now, the amber pieces that have a hell ant in it with another ant is actually a different ant species.
Oh, huh.
And so...
Interesting.
I've argued like the possibility of there being like that they're just a slightly asocial ant is there's no reason to suspect that that's not the case, because there isn't any evidence in terms of the amber where there's multiple individuals together with reproductives as well, right? Because you could argue that if there's multiples together, maybe they were having a territorial interaction.
Sure.
Right?
Yeah.
And so it's it's not great for in terms of the evidence for that.
Still, I mean, that's often how it goes in paleontology, at least. So that's fair.
Yeah. But I mean, I think
I can relate. Yeah.
It's not that you can't get ants. Like, that's how we actually identified that ants were eusocial, is because there was resin that caught a ton of ants together with reproductive, and it was like, okay, great. Like, that's awesome. We now suspect that they're colony living. So that is how we can identify something like that. But if you can't find the fossils, right, then it's very much like, I hypothesize this might be the case without necessarily having the evidence for it.
Makes sense, yeah.
But no, I know I say jokingly that you caught me, but like, that is just a fundamental piece of the science.
Sometimes you just have to come up with, like, what is the most parsimonious explanation for this with as little evidence that we have, and then just, you gotta be content with that sometimes.
Yeah, and I wish that there would be more, but there's just not enough, like, data of the resin being collected. But I guess for this paper, it didn't end up mattering because they were looking for variety and numbers and not taking anything into account of their specialization influencing species number, or like how they're group living, anything like that, versus, it's just diversity. They just like, this is a different species, but it's an ant, and this is an ant, and so on and so forth, right? Which I think it makes sense that they're not overly jumping. Although they did say in the future directions they might be able to interpret, using this kind of data set, evolution of behaviors as well. Because you've tied it here to this, the angiosperm revolution. But why can't you tie it to something else? And I like that mindset. I think it's really cool that they can have that hypothesis of like, well, we have this fossil data with your modern data. All you're basically putting is different factors on the timeline and parsing the data in different ways. At least that's my naive take on what else they can do.
Also, for chat, if anybody's just waltzing in or anything, you might be wondering, like, what is the angiosperm revolution? And this is something that we talk about as dinosaur paleontologists all the time, because it happened during the age of dinosaurs, in the early Cretaceous period. But this is when flowering plants first evolved. And today, in our world now, most of the plants that you're going to encounter outside are flowering plants. That's fruit trees, that's, willows and grasses and roses and, just about everything. Except for, like, it's palm trees as well. It's basically everything except for conifers, like pine trees, and ferns and cycads, like we're going to be talking about later too. So it's the vast majority of plants around today are angiosperms, and they did not exist up until the early Cretaceous period, about halfway through the age of dinosaurs. And as these plants develop, they just change the landscape entirely. And and they probably had a huge influence on the animal communities living there too. They may have heavily influenced dinosaur evolution. We see the rise of the iguanodontians, of duck-billed dinosaurs, and ceratopsian dinosaurs, the horned dinosaurs like Triceratops, only really show up after the evolution of angiosperms. So it was hugely important. Yeah.
Yeah, and I guess it had
I've got cat hair all over my face. I gotta make a washcloth and keep. I'll be right back.
Yeah, of course. Y'all, I think Moon Pie really likes Danny, and Sweetie Pie, and they might be getting up in his face a bit doing poor Danny. But yeah, y'all, right? Just before Danny comes back, some of the hypotheses that they were putting forth is that how this revolution in the angiosperms, these flowering plants, can lead to diversification of ants and also losing them. On Tiny, Charlie's Dragons, the trapjaw ants. I don't know if you'll see this video, but when they're closing their mandibles on someone's finger, they can't actually cut you. If they hit your finger, they bounce backwards. So you're totally safe on that front. Don't worry, Charlie's Dragons. The trap jaw ants, Danny, with the horizontal mandibles. It's one of the fastest movements in the animal kingdom.
Uh-huh.
Super tiny, right? Well, I guess not like a picnic table ant. There may be like, length of like your thumbnail. And one of the fastest animal movements that they launch on your finger, but they won't cut you. They'll actually just launch their bodies off into a distance.
Because your finger to them is an immovable object.
Yeah, so but they'll try.
That's really funny. Oh man.
There's a vampire ant that's called that does the exact same thing, and the vampire ant, we think, not only does it for prey catching, but also launching their bodies off into for protection.
Gotcha.
And yeah, 6 foot ant knows they're ants. The army ants, there is the soldier caste of army ant that has these really large curved mandibles, and those are for protection, and primarily going after leafcutter ants.
Interesting.
Because they actually will burrow underneath a leafcutter ant nest and pop up in the embryo chamber and start stealing babies.
Holy cow. How do they know where to? Do they just kind of scattershot go everywhere, or can they hear, or how do they manage to pull that off?
It's this weird nomadic type of ant species that just patrols on the jungle floor. They have like a home base where the queen is settled. They go out and patrol. They just get a bunch of food, come back, and then a few days later, the queen packs up with them and they go a new place. And so there's some cues that signal them for underground as well, like loose soil, like as a nest site. But what's weird is, and this is a behavior no one really understands, is that the soldier of leafcutters will stand out, and they will antenate or touch antennae with the army ant soldiers, and they'll be at a standstill. They'll just be like antenating each other, and no one's moving.
So
and that's kind of like this like battle, like we're ready for battle, but we shouldn't battle because we're on standby. But if someone makes a move, it's just carnage.
Wow.
And what's really interesting is if you do that, if you put a carpenter ant in, the soldier leafcutter ant will try to do the same thing. But the carpenter ant is like, I'm not evolved for this. I don't know what you want, and will run under the legs of the soldier, go steal the baby, and run out.
They're not playing by the same rules.
Yeah, and it's because, it's like evolution hasn't acted on that behavior, so why would they? But it's just, it's interesting.
Yeah.
Well, what are the signals that happen? And like, no idea. It's just a phenomenon that's been recorded. Like the poor leafcutter soldier's like, hey, we're... Come on.
Yeah. Oh man, that's funny. Interesting. Really interesting.
Yeah. So I was just touching on the theory, like some of the theories that they were doing, and actually they had all 4 hypotheses were, validated in some way by the data. And so just a quick look, Chad, of what the data actually means. During the time of this angiosperm explosion is shown in green. Time is on the bottom axis. On the y-axis is the diversity of these ants. So higher means there's more different kinds of them. Lower, it means that there's less. So it's a really nice, I think, straightforward way of reading the graphs. We don't have too much data. Like, there's no numbers on here. The numbers are actually in the raw data set on all the supplemental figures. And it's just like a nice, clean, easy way to interpret what's going on. So here, in their first hypothesis, they suggest that, okay, with too much specialization, you see a decrease in the diversity of those stem ants, those ancestral state, and an increase in extinction. Which is actually that's like the baseline hypothesis that we found. But you can see here, like, they're just trying to establish that this is a phenomenon that happens. For the later data, that will be important. And then, so this is one that they had too, which was the competition hypothesis. This is the one that kind of ended up being disproven. The idea being that these 2 ants, your modern ants, which are, well, again, like Danny explained, the crown ants versus the more ancestral state, the stem ants, were going against each other at the same time. and outcompeted one outcompeted the other, and that's why you saw the rise of crown ants and the decline of stem ants. And if it is lined up to the angiosperm revolution, then you'd see some kind of connection there as well. But the more likely experiment that we see is this one here. So the blue is the increase in angiosperm plants, different plants and populations and types, and the green is the increase in the diversity of just global ants in general. So this is not parsing out the hell ants and for future extinct ants and the crown ants. This is just putting everything together. And what you're seeing is, even though there is an extinction event that's happening, that's shown in this upper graph with the stem ants, you're growing at a near exponential rate of your crown ants, which are your modern day ants. And it's connected to the increase in angiosperms. So this, to me, was the coolest figure, Danny, because it was kind of like, even if we take into account extinction numbers, you're having a huge increase in diversity over time.
Right.
And it's linked to the blue line, which is the increase in angiosperms.
What I'm wondering here is how well constrained stratigraphically all those fossil ants are. Like, because a lot of these are going to come from amber, and we don't often have firm dates on a lot of these amber deposits. It'll be like a range. It'll be like, it'll be within this 10 million years here. And I guess maybe that's why the line is so smooth like that.
Yeah, there is.
They're kind of averaging it out.
Yeah. Let's see if it was in the main. I think it's buried in the supplement where they had a little bit more of these ranges plugged in, and so you saw it's a little bit more messy. But I think with 24,000 range
you can get
numbers,
an overall trend and make that smooth line, and it'll comport pretty well with reality.
Yeah. No, you're right. Like usually, like we can't date the resin. And so you have to get that strat. And like actually how you were saying, Danny, of like if you find something, leave it there. Right? Because the paleontologist needs to identify the location, where in the strata it is, what that means for the age of the sample, etc. And a lot of these resins, I mean, they went into it the way that we've gone too, is that there's the Baltic amber is in the rough 50 million years old. The Myanmar Chinese amber in the 90s, 100s. The Dominican amber is in the 30s, right? They gave those same ranges that we talk about, and it's like, well, you can't, if you're just getting them off a personal collection, which is what a lot of it was, you're not going to have that strata information. You just have to go off the average range.
Exactly. Yeah.
Which, not great, but we work with what we can. I still think, though, like it's, with the numbers, even if there were some individuals that didn't go in the same trend, we're still seeing a lot of what might be. Like with so many individuals, that global trend is important to note.
Yeah, for sure.
Yeah. Farf, welcome in. How's it going? And then the final one, this buffered extinction hypothesis. So for this one, I'm actually going to read what they said. Diversity of ants increasing synchronously with angiosperm diversification and extinction decreasing oppositely. So the overall extinction rates, if we measure them numerically, are going down with time because there's so many new ant species coming up. And so and what's cool is too, there's a shift in these lines. So if y'all look, each of these graphs is actually plotted the same way. So the start and end times that they're plotting are the same as are the location of the green bar. And so you can actually see that cross between the two is shifted. So here is the intersection point. It's further along in the angiosperm revolution, and it's kind of I wish they would have done an overlay that corresponds to that increase in the angiosperms like they plotted in this blue line, because it really shows that interaction between those two. So yeah, I just thought it was cool that they were able to grab these kinds of rates and match that, the extinction levels overall are going down, and that's tied to this diversification, which is tied to the angiosperms coming through.
It's really neat stuff. Yeah.
And yeah, the other papers we're kind of looking at the other figures we're looking at more particular ant species and ant events. So they were looking at net diversification from different data sets without single points, and this G through I is particular time of late Cretaceous. So then they focused on particular times. I don't know if they're like they talked about the Cambrian, the KPG boundary, which I don't know, Danny, if you wouldn't mind refreshing folks on why that boundary is important and why maybe they were looking at it in this paper.
I think apart from the Angiosperm Revolution, the KPG boundary is kind of what helped shape our modern world in a lot of ways. That's when that giant asteroid struck the Gulf of Mexico and caused the extinction of all of the dinosaurs except for a few species of birds. Most of the mammals, most of the terrestrial reptiles, all the flying reptiles, ammonites in the sea. Basically, like, between 70 and 80% of species on Earth died out at that time, give or take. That's the KPG boundary. KPG stands for Cretaceous Paleogene. It's K-PG, for anybody wondering. So yeah, it's the end of the age of dinosaurs. And a bunch of plant species also went extinct. It's like 40% of plant species or something like that. But the angiosperms were kind of the big victors afterward. Like they'd already been kind of taken over the world, and they kicked them into high gear after that. But yeah.
And one thing that's, I think, really neat is if we look in, so this black line here, for example, is looking at that ant diversity. And so if you look at that KPG boundary, it's like right beforehand they were increasing in terms of ant diversity, and then you just have this huge sustained, like, continued growth after, instead of that species die off, like they're still doing really, really well. And then like there's like ups and downs, right? But it's still right after that event that's supposed to be this big event that wipes out a lot of things, like they're doing really well.
Yeah. It's interesting to think about. Like, why do ants do well across the KPG boundary? And a big part of it might be living underground. Because is it... you know more about the fossil history of ants than I do, Bullen. Is it thought that crown group ants were already living underground at this time? Already digging burrows and stuff like that? Like having their colonies subterraneously?
Yeah, so it looks like your leafcutter ants, which would be outside a little bit more and being caught, were about 30 to 50 million years ago, depending on where you're collecting.
Gotcha.
Okay. And then there's still, like, you can get relatives of, like, carpenter ants in the time that 90 million is, but you're getting them in groups. At least you're getting them walking on the outside and doing collective group behaviors. In terms of underground, I think there are actually more... They started living in wood, even in the 90 million time. And that's why we're able to collect, I think, a lot of those colonies. So I think it was Hanny that asked if there ever a fossilized ant nest. And so there's big remains of ones that lived in trees where it was the amber essentially just coated everything on the inside. But then underground, like... that hasn't unfortunately happened, Hanny. So, like, that's how they would be preserved. There might be imprints of ants, like, that we found in stone, but, like, getting a whole colony, like, it's just... the likelihood is going to be super, super rare to have anything but just a few. So you'll never actually get the colony. Actually, even nowadays, to preserve an entire colony, like, pouring that molten aluminum...
Yep.
that gets you structure, but not really individuals. And so, like, I mean, like, if you were saying, like, lava flow or something, like, I think earlier in the chat for a preservative, like, that might get you structure, but you'd have nothing to know it's actually an ant. Like, you'd be guessing, like, maybe this is an insect, but you wouldn't have a bug in there. And so then... right, what is exactly that's coming from there? So, yeah, Hanny, unfortunately, no. I think living in trees... and, like, close to the source of the resin where they'd be preserved started happening about, 90 to 60 million years ago, really heavily, where we have that kind of fossil remain.
We had some good questions in the chat, too. Psychanalyst wanted to know, what's that spike in Eocene diversity? If you go back to the graph that we were just looking at.
Yep.
You zoom in. Yeah, that might be the end of the Paleocene-Eocene thermal maximum. So, like, Earth temperatures just throughout the Cretaceous, they just, or at the end of the Cretaceous, they're just kind of getting warmer and warmer and warmer, and then Earth is going through this period of like massive global warming where you've got, you've got palm trees and tropical foliage growing in the north slope of Alaska. There would have been zero ice on planet Earth at this point. And that it gets warmer and warmer throughout the Paleocene and the Eocene epochs, and then temperatures drop. I think it has something to do with Antarctica finally separating from South America. And so suddenly you get these, you can get this cold water rushing in between there, and then it just kind of disrupts the Earth's climate system, and then it suddenly cools. And you get the rise of grasslands. That might be right there. That might be grasslands opening up. Because I'm guessing that there's a lot of different ants that really love grasslands. I'm guessing.
And then there might be the associated prey as well.
Yeah.
Like that's I think the cool part to think about too is how we can't view them as insular. Like it's not necessarily just like one organism doing really well, it's also dependent on others. And I think that's one of the key points of their paper is that most of the ants that we have today are diverse in what they can eat. So it's not just one prey population that had to do well, but maybe like a bunch of insects are doing well now. Like I think the rise of beetles is actually also tied pretty closely to this angiosperm explosion, and so you see a lot of beetle-like creatures that are prey of ants, and so it'd be actually cool to see the two overlapping to see if they're running after each other in terms of like diversification. But that could be something too then, like their food sources are also expanding.
Mm-hmm. And we had a great question from Hanny's there too. That ties in with beetles also. Were flowers pollinated by bees back then, or had they not evolved yet? I'm pretty sure bees start to evolve. Like, we get pollinator-type bees, or bees that we imagine being capable of pollinating flowers, around the same time that the first flowers show up. But we also had a lot of beetles, too, that we think were pollinating flowers. What's your understanding of that, Bullen?
It's the same, yeah. And then the way that that's been identified, too, is there is amber with pollen patches on the individuals, and so that's the hypothesis. Like, even if it wasn't intentional, like they're not active, like, and no insect is actively pollinating, but they're just the pollen's hitching a ride as like they're getting the nectar, right? And so that's the thing is too. It's like, why would this beetle be climbing into a flowering plant, like maybe getting the sugar from the nectar, but then also it would be pollinating other individuals. And then bees too, Hanny. You had a lot of stingless bees, so they don't look like our modern day honeybees, but they might look very different. Like, think of your local pollinators. If you Google like wherever you live plus local pollinator bee, you'll get a lot of these really beautiful stingless bees that are much more reminiscent of that ancestral state and what they might have looked like. And I know before I looked at them and knew that the thing was, I was like, I don't know what these are, but then you look and you're like, wow, there's such a diversity of them. And yeah, there's solitary bees as well, as Sai's mentioning.
Where do hoverflies fit in there? Are they a different kind of insect entirely? Are they flies, or are they hymenopterans?
I believe they are dipterans because they have these haltear structures. So that's the tell. Like, there is this little nub that comes off like right around the armpit on either side, and it's either, depending on who you talk to, it is for mating, but yet there's, if you chop it off, they still mate just fine. So is it really for mating? And then some studies suggest it's for flight balance, but there's some of these dipterans where it's already kind of rescinded. So if you remove it, they fly no problem. Versus those hoverflies have much larger ones. And I don't know if they've been tested for flight stability, but that's like a clear something as well there. Yeah.
We would get these. I think they were hoverflies in the field when I worked up in Montana in the Hell Creek Formation. My crew chief called them sweat bees, but I don't think they were bees. I think they may have been flies, but they looked like they were trying to mimic bees. They were black and yellow with stripes, and they like kind of flew around, and it almost seemed like they were trying to act like bees, but I don't think they were bees. I think they were related to hoverflies or something like that.
Yeah, it's cool to see the the overlap in colors, right? The mimicry that you see, and then also when you get to bee behavior or mimicry, it's just like, well, what is this for, right? Like, is it for some kind of I assume like defense, because that seems to be predation's always a good driver for things. But then is it also maybe to trick the bees into like giving them some food? You get right, like the motives of what's happening is really cool. Eavesdropping is also
pretty opportunistic.
Yeah. Eavesdropping is also considered to be a possibility for some of those, where it's if the bees see that it's similar behavior, you know, the other ones will be allowed to follow, like the mimics, and they can get some of the food sources too.
Gotcha. These ones would fly up to us in the quarry when we're working on a really hot day, and they'd go and just like drink a little bit of your sweat, and then they'd fly off. That's why Denver called them sweat bees. But
there is actually a group called sweat bees as well.
Oh, there is?
Yeah.
Oh, okay. Interesting.
We've looked at some in amber.
Uh-huh.
Let me see what the actual definition is.
Are they hymenopterans or are they flies, the sweat bees?
They're halted bees.
They're bees. Yeah.
So they are... Yeah, it's these like really pretty...
Ours look very different.
Oh, okay. Yeah. Gotcha. So they may have actually just been the mimics there.
I think they were. I think they were some kind of weird hoverfly or something like that. But they were all stripy, and they didn't. The body shape looked very different from a bee, but the coloration and the way that they kind of carried themselves, it felt like they were trying to mimic a bee.
They say that there's something really similar as a cuckoo wasp, but I think it might be similarity based on like the imagery that we have here versus the behavior of it. Yeah. Yeah, the sand wasp, and there's a lot of diversity in these. But yeah, the sweat bees are the ones that we find in the amber that have this reflective prism pattern almost on their bodies. It's like that structural color that we're seeing. That's like one of the fast ways to identify it is, and then the antenna to accompany with it.
Neat.
Yeah, so I thought again, like, it was a really cool study. There was a lot of unique data points. This one too, this one I think probably takes the cake. The number of lineages. So, just for pulling out from what they say in the figure, let me grab this panel I. Let's see, so they're looking at net diversification that's increasing during the late Cretaceous, Eocene, and Oligo-Miocene era. And so this is, I think, the cool one where it's like they're just looking at overall ant lineage number.
Going up. Yeah.
Yep. And so this is their argument that it's tied to that angiosperm revolution, where you see at the beginning of the angiosperm revolution, that green bar, it's low still because the animals need to start adapting to living like this, but then it starts rising after the KP boundary, and it just keeps going up and up in terms of diversity. And so their argument was, without this diversification, the plants happening, you probably would have lost your ants, because they were like the ones that the stem ants that exist, they were so specialized that there was no way that they were going to survive. And you can kind of see that with like here, these overall numbers at the beginning are pretty low, relatively speaking. And so it's like the number of lineages here, like it's not in the hundreds low, it's like, under 50. And so is that expansion allowing the animals to survive, which I think is, again, really cool. And like there's so much data in these figures, y'all. It's really, really impressive to be able to pull something like this out. And then finally they just have it's called the paleo environmental correlates and potential drivers of diversification. And so each one of these is looking at how much the increase or decrease in diversity of the animals with different stressors. And so it's a little bit back and forth, unfortunately, on the figures, and it's hard to zoom in, but they have pictorial representations of what could be driving this. And so like here's all the things that they were looking at. So spored plants, sea levels, continental fragmentation, gymnosperms, the angiosperms, carbon dioxide concentration. Um, there's so many. And the big takeaway is the biggest driver is these angiosperms. So I thought this was a nice way to kind of summarize up the figure that it's not just from flowering plants that's driving this, but rather it's from a lot of different like it's that one factor of flowering plants and not just like all these other potential things that could be happening. Like you said, Danny, the temperature changes. If that alone could explain diversity, then this is a moot point and we don't really care about the angiosperms.
It's really cool because they've essentially, it's like they've done a bunch of potential rebuttal papers and said like, oh, here's why all these potential rebuttal ideas don't work. They tried a bunch of these different methods, a bunch of these different variables, I should say, and tested each of them if they're responsible for the rise of stem ants.
Yeah.
Or of the rise of crown ants, excuse me.
And that
and the decline of stem ants. And yeah.
I feel like that's how science should be done.
Yeah, absolutely.
Right, for really, hypothesis really cool. Driving, like, because like you said, you're the expert trained in the field. And you could have told me a lot of different, and we have talked about different things happening at different points in time during the paleontological data. And like you said, temperature, or maybe the reason there's diversification is because they're all in a landlocked area and things split, and that's what allowed them to expand. I don't know, right? That's why not? That can be a perfectly valid hypothesis. And so it's just really cool to see that that's not really the issue. So like here in the figure, y'all, is kind of a mess because they define in each panel something different. So blue in some of these panels is blue is origination. In AC, E, and G, and extinction is red for events. And then here is like the breakdown, which is kind of frustrating to go back and forth. But they're looking at, again, global temperature, continental fragmentation, sea level fluctuations, global variation of atmospheric CO2, the ferns, gymnosperms, spore plants, and angiosperms. And so the big thing that we're wanting to look at is this icon here, which for those of y'all who want the paper, again, it's on the Discord. But this is their angiosperm image, I guess. And that you're looking for increases in blue. So higher levels of blue means that there is increased diversification, and then the opposite direction moving red would be decreases in extinction levels. And so it's kind of tying those two box plots together, where increasing in diversification that also is a fundamental decrease in extinction. Because if they're both coming upward at the same time, like this is a good one to look at. See, you have an increase as a function of the diversification of the angiosperms and a decrease in extinction. If both were going up, then everyone's dying out even though they're diversifying, and it's kind of a pointless effect, right? That's not to say that there are other factors that could be acting on this. So like here, this is the continental split. We see there's an increase in diversification but no effect on extinction levels, and that's probably because as things were splitting, you got rise of different environments, and maybe there was more species of prey that were starting to live in these different areas that were fundamentally different, and that could be a driver for diversification. But again, it didn't affect extinction rate. So it's kind of cool to see all these differences and how they're acting in. But I think, again, the biggest baseline, biggest takeaway is this angiosperm driving so much of the diversification and negative of extinction levels.
It's a complicated figure, and you're doing a much better job explaining it than I could. I'm wondering, though, not to put you on the spot, but if you were to try and design a figure like this, Bulent, to portray the same information in a more intuitive way, where would you start?
I don't like these bar graphs, just in general. Especially with so many different variables. So, like, some of these have the singleton datas, and they're looking at only paleontological occurrences. And then here they're looking at paleontological and modern occurrences as well. I think I like having it split. I'd also add in a third category of just the modern ones. And that should be your negative control because you're not plotting against anything, right? There isn't any extinction drivers. I would have some kind of... I mean, kind of like these graphs. I think these make a little bit more sense, right? There's time as a variable.
Yeah, for sure.
Like, you can have time in. You can mark when some of these big events happen. Like, I would like to know the resolution of that they have for tectonic breaking, like the landmass is breaking, or, you know, put in, like, you said that maybe the spike here is due to temperature. Well, that's great. Like, add in on this figure. Don't just have the variable temperature, but have what that temperature range is.
Right.
And so that's how I would graph, like, here's the plot of how the, you know, the variable of extinction levels and diversification happen with this one variable of temperature. And then you should be able to see if there's a correlation or not. And so, sure, it might be a multiple graphs, but I think looking at them individually versus, I don't know, I have trouble looking at these, especially when it's also pictorial, because I don't know about you, Danny, I couldn't automatically tell that that's an angiosperm.
I couldn't either. I don't know. It's like a Rorschach test. Yeah.
Like, I guess I can see that that's a little seed and that's maybe the root. Maybe. I don't know if that's what I'm looking at. To me, it looks like a fruit fly larval brain is what I see.
Yeah,
and, like, because that's what they look like when you dissect them. I'm like, all right, that's clearly not what it's meant to be. But the data's great in it. Like, it's a really cool data that's coming out of it. I just think it could have been...
For sure, yeah.
A little bit more accessible, and you already have the data for the sea levels and the continental plates. I think, you know, you asked me, can you make it simpler? And here I am like, well, put in these variables and what the difference is what. But I think it would make it easier to understand because you'd be looking at each event isolated, and then you could look at the graph and get what the information is from, like, the temperature perspective. And, you know, the rise of fall of gymnosperms, rise of angiosperms overlaid on these two variables that you're looking at anyway. And so you can choose to ignore that. Like, you can just look at, okay, overall temperature didn't have an effect, move on. Or you could be like, well, let me see how the temperature affected the lack of correlation, let's say. So that's how I would go about it. I don't know, Danny, if you would, like, what would be common in paleontological papers for this kind of data even?
It's weird to see bar graphs like this, especially, like, coming off of a central axis. I don't know what the name of this kind of a graph is, but
I should know this. Lita's going to be very mad at me. I don't remember.
I would have no idea. So
I think it's...
so I'm not trying to get you in trouble with Lita.
No. It's similar to a... I don't think it's just a straight up, like, vertical bar graph. I wish I knew what the
central axis.
Well, because this could be fold change, or like instead of raw numbers, and so I would. It would be easier to delineate if we knew what that lower variable actually stood for. Versus here, it's just. Oh God, I don't even know if they tell us in the figure legend.
I've been looking for it and I don't see anything. Yeah.
Shrinkage weights. I don't know if that's actually what they're going for. It must be some kind of magnitude measurement.
It says Bayesian inference of correlation parameters, so there's something interesting and statistical going on there, but, like, it's sailing over my head. This is not my kind of...
It's not my wheelhouse either for stats, but I assume it's like orders of magnitude, and so, like, stepping from 1.5 to 3 isn't just like a linear chat, but it's like a much bigger jump that you would have from, like, an earlier point as well. What's the benefit of this? Oh, so Nelf, this is an aesthetic rule that I know nature has put in, where if you have bars that go under and go negative values, they don't want it to be your standard vertical bars. They want it to be like in this horizontal metric, because it's misleading because it's not an actual negative value. Like, you're not losing diversity like you're plotting against a measurement of magnitude, and for that you have to have this type. Like, that's some of these journals, like the PNAS, Nature, Science, have like some rules now of how they want. Even like, you can't have, for example, now vertical bar graphs in a lot of journals. They want what are called box whisker plots, because those encompass the actual data points within the bars, and it will show you outliers as well, so you'll get more information out. So I think this is a means of standardizing like fold changes as well. Claire does not like it, Danny. 6 foot ant doesn't like it either.
Yeah, it's not my favorite either, Claire.
Yeah, I would have preferred
the
line.
It's really, really cool.
Yeah.
I mean, like, the science in this is really, really... Here we are griping about the graphs. That's my fault.
Raiders, welcome on in. Welcome in, Hagerman Hour. Welcome. I hope you had a lovely stream. What were you doing? What were you up to? I see there was some game development. Welcome. Let us know what you were doing. For the Raiders coming on in, my name is Balint, 1/3 of Science Streams. The other 2/3 are my wife Lita and our daughter baby Alona. We're research scientists, our PhDs in molecular biology. Do live content here on Twitch 7 days a week, sometimes even 8, as Danny says. And this is Professor Danny Anduza, single greatest science communicator on the internet. His name is below in the episode description. Make sure to click his name and follow him. And today we're chatting fossil science, genetics, and more. Reviewing 2 papers. Raiders, welcome in. I hope you had a great stream. Let us know how it went. Sorry to cut you off, Dan. You were saying, we are griping about the figure aesthetics, but the data itself.
Science, this is so cool. Like, they've had so many different variables that they explored, and I feel pretty confident, I guess, in the results of this paper. I feel like they did a really, really good job of exploring these different possibilities, and the rise of angiosperms seems to have the biggest effect on ant diversity.
Yeah, and it's, again, I feel good about the data because there's so much put in.
Yeah.
Like, there's so many samples that they looked at. Like we said, Chad, 24,000 extinct fossils, like prehistoric fossils, and then 14,000 modern day specimens, just to be able to get these kinds of lineage graphs and all the, like, it's a lot of data. And it's kind of neat that it, in a way, validates this original paper in a way, that there is a link of diversification tied to the age of angiosperms. Like, this, here was like, oh, we think this is kind of a thing. Here's a couple of, phylogenetic trees of this is like the modern ants here, right, that they had phylogenetic trees for, and then they had a smidge of ancestral ants. And then here, I mean, it's kind of a similar graph, right? There's the range of the increase in angiosperms, and that ties to an increase in ant species, and here we go. There's the paper, but it left a lot to be desired, y'all. Like, there was other hypotheses that could have been driven. There's other factors that could have been. Are you cherry-picking just a few fossils, and that's what you're getting this result from? Were they in competition with one another, and one outcompeted and killed off the others? Like, those were popular hypotheses. Even, like, I guess up till this paper that we're reading now, like, it was still not disproven. It wasn't a popular theory that the crown ants or your modern ants were competing for the same resource. But, I mean, it wasn't disproven. And this is the closest that we've gotten to it. So I think it's really, really beautiful.
It's good stuff. Yeah, this is a really neat paper, and I think it also just kind of emphasizes how important the rise of the angiosperms was. I mean, this is like... I like this kind of newish phrase that's going around, the angiosperm terrestrial revolution, I think people are calling it, ATR. Because it's like, this is the landscape, the whole foliage just totally changes during this period. Like, angiosperms today are the majority of plants that we see around us, and they just did not exist before the early Cretaceous. So, like, having the base of the food chain like that in terrestrial ecosystems completely change not completely changed, but changed so much. That's a seismic shift. That's big. That's big change, and we see that reflected in ants. And I'm curious to see. I wonder how you could devise an experiment to see how this actually affects the rise of different dinosaur groups. I wonder if you could use kind of the same methodology here that they used for ants, and then apply it to the evolution of dinosaurs in the Cretaceous. I wonder if we have enough specimens, if we could get results as good as these. I kind of doubt it.
That's what I was going to ask you, is that if you thought maybe the limiting factor, like almost the short pitfall of this model, is that it required so many individual specimens to really get a good resolution.
Yeah. I mean, we've got so many more variables, I think, that might, well, I don't know. There's a lot standing in the way of us just having tons and tons and tons of dinosaur specimens to be able to test. We do have a lot, but there's a lot of preservational bias. There's a lot of possible synonymization, like, are these actually two different species of dinosaur? Or is this a juvenile and an adult of the same one? Stuff we've talked about a bunch. I don't know. I look at this paper and it makes me a little bit jealous. Wish we had this. Wish we could do this for dinosaurs, you know?
It's
so cool.
I mean, I guess it was neat, too, that they pointed out limitations of the study, though, right? I think that's
for sure.
one thing that's kind of lost in a lot of papers. And so, like, you know, how they're pointing out that the fossil record may be biased because of their biology and morphology or geographic distribution. And I thought that was a really cool thing to point out, because that was one of my first thoughts, too, is maybe they just, you know, are selecting for ones that are really well preserved. And that's because of the paleontological data, and that might be something like we talked about it. Hanny pointed out, right? If they're not getting well preserved because they're underground, I mean, you're inherently selecting for ones that are being well preserved because they're not.
Biased, yeah.
Yeah. And it was cool, like, you know, they're like, you know, we can't overcome it now, but there's new discoveries that could be coming in, and, you know, that's like a nice point that they have. We can always update the model. The geological periods around the KPG boundary exhibit a paucity of fossil ants, and the pattern holds true for many insect lineages dating back to the Cretaceous. And so they need to, you know, like, we need new fossil deposits. And I was wondering, Danny, do you know why that might be? Like, what about that time?
There's not very many places around the world where we actually have the KPG boundary preserved. In a terrestrial setting, it's much more common in marine settings for whatever reason. Maybe because sea levels were relatively high back then. But yeah, we don't have that many spots where we can look at what animals and plants are doing right as the asteroid strikes, like immediately up to that point, and then in the immediate aftermath. We don't have a lot of strata that are bounding the KPG boundary like that. So, like, we don't really know a whole lot about which birds were existing at the time, or I don't know. For dinosaurs, we've got to look at, like, the Hell Creek Formation in Montana and the Dakotas and Alberta. Yeah, it's... Maybe I used this analogy last time, but it's like you're trying to figure out what's going on the other side of a fence, and there's only a few little holes that you can look through, and the Hell Creek Formation is one of our biggest ones, but, like, we gotta find some more. I can't think of finding, like, a single insect fossil in the Hell Creek. I don't think I found any insect bits in the Hell Creek during all my years working there. No body fossils. I think most of what we know about Hell Creek insects comes from bite traces on leaves.
Oh, wow. That's a...
So, yeah, I don't know if we actually have any insect body fossils from the Hell Creek.
Huh.
I think almost everything comes from, you know, bite marks on leaves. Yeah.
And that's just like a big inference, right? Because you're not having actually any body. You're just having... You're comparing to, presuming, right, modern day bites and and seeing, like, what they look closest to.
Exactly. Yeah. Which can be pretty good, but well, I don't know.
It's not the worst kind of evidence we have as paleontologists, so put it that way. Yeah.
Nelf says
Yeah.
that in reality, I would think that we've only discovered fossils of only a small percentage of animals from the 40 to 100 million years ago.
Overall, that's true. Yeah, yeah. But there'll be certain groups that are overrepresented because they leave a lot of hard parts. So, like turtles, we've got an excellent record of turtles because, they've got this big plastron and carapace, and when they die, it kind of, I was going to say explodes, but we use this term a lot. It doesn't actually physically, there's no fireball or anything, but, it scatters. The pieces go everywhere. It explodes in that regard. And just finding a single piece of a turtle shell can sometimes give you enough information to actually identify what genus or even species it's from based on a single fragment. So, like, the turtle fossil record is excellent. The fossil record for, say, slugs, not excellent, because they don't have a lot of hard parts that fossilize. So you get these certain groups that are overrepresented in the fossil record and others that are underrepresented or absent from the fossil record just by virtue of their biology and their morphology. Yeah.
And so that, like, lack of discovered fossil, if we've only gotten a small amount of fossils that we could discover, there's even more that just didn't get fossilized or are so rare that we may never find, and that also, isn't necessarily accurately represented in what we do end up collecting.
Definitely.
Yeah.
Yeah.
Oh, Socks had thoughts about that part. You have thoughts?
That was audible. Wow.
Hi. Yeah.
Aww.
Yeah, I know. She had a lot of thoughts. I tell you, this cat, her favorite thing is to sit in this chair next to me with her blankets, rest all day, and then come over for snuggles, and then go back to the chair, and then have, like, that's... just living her best life. I'm like, all right, I'm good with
Good
it.
for her. Yeah.
And yeah, then the final, like, kind of caveat that they give is if there's something wrong with the model in terms of how it's processing, like, where things fall in the evolutionary tree. And it's such a standard thing to be a potential pitfall, but it's really cool they mention it.
Yeah. I wonder, in hearing about, in reading that the authors are listing the limitations of their own study, do you think in any way this is an effort to try and get it through review a little bit faster so you don't have a reviewer too going, well, did you consider this or that? Like, if you just put it in your manuscript that, like, yeah, we have these limitations here, and you just say it with your chest at the beginning, is there a higher likelihood that they'll leave you alone about that, you think?
So, this is not my thought. This is what I have been told. Is that when you're submitting the initial paper, do not put anything like that in. Because if you do, it's a target that the reviewers will right away hone in on, and that you're painting a target on your back to address it. And so one possibility, maybe they were really honest, and they were like from the get-go, like, these are some limitations. Because the other thought I've had is if you put potential shortcomings in that you can answer later, like let's say they put something in and they were still working on it but they wanted it submitted, and like this could be a shortcoming, and then the reviewer is like, hey, wait, this could be a shortcoming. You're like, oh, I've already done the experiments, now we're not in a year of purgatory.
Yeah.
But more than likely, I think the reviewers probably fired back with like, hey, there could be some problems here with your model, and they'd be like,
Yeah.
you're right, we're gonna put in these potential caveats, and that will demonstrate as to why there might be some potential pitfalls. Now, again, naively, I want to believe it's always option 2 where they just put it in right away because that's being the most honest, but chat, like, it's publish or perish, right? If you don't get those papers out, you won't get funding, you won't get that tenure position, the student won't get picked up for a postdoc position or what have you, and so you have to put out papers. So more than likely it's the first option where it was like, here's how everything works, and then the reviewers may have been like, well, what about these things? And you'd be like, well, that's kind of outside the range of what we're able to do, and so we'll put it in as a potential pitfall and limitation. Well, how is it normally like for you, Danny? What have you heard?
I don't like. I feel like just naming it at the outset sometimes works a little bit better, but I don't honestly have enough experience with this kind of thing to be able to really draw any firm conclusions from that. So I was really eager to hear what you had to say about it.
For picking up, like, if you're actually digging out new dinosaurs, are there standard things that you have to account for and put in something like this? Like, I guess that's what I'm trying to get at, is like, is it...
Depends who the reviewers are, you know?
Okay.
Yeah.
Hello there.
Yeah.
Hello, history buff. Welcome in. That's. It almost feels a little bit more honest.
Yeah, and I think. Yeah, it'll also. A big part of it too is like this sort of study that you're doing will dictate which journal you submit it to. There's a lot more of that I think in paleontology than there might be in like genetics. I'm guessing maybe. Where it's not like, oh, what's the quality of the research? It's like, what are the qualities of the research? Like, is it just a descriptive paper? You might send it to the Journal of Vertebrate Paleontology. If it's something that's a little bit more, like biology oriented, you're actually looking at, I don't know, numbers of species over time and their evolution. You might send it to Journal of Historical Bio or PNAS or something like that. Yeah.
That makes sense. I know for us, it's for genetics observational now, which is wild to me because 15 years ago this would have been a science paper, is a sequencing experiment. Like if you get a data set of like, here's all the transcription factors that change during this stressor. If that's all you have, that's a specialty observation journal.
Interesting.
So like, even though it's
which is pretty low bar to get in, right? It's just quick and dirty. Like, here's what we saw.
Yeah, like here's almost a data dump, versus like the higher tier ones would be, okay, we identified these top genes. Here are the candidates that we had. Here's why we think they're candidates. We tested them. We made a mutant, and like, 40 figures later, you have your paper in Cell or Science or something like that.
That makes a lot of sense. I think like if you just find a new fossil, it'll go into like one of your more like workaday journals like JVP or something. But like if you find something that you think has big ramifications for the biology of a certain group of dinosaurs, or it increases their range tremendously or something through time, you might try and get into a higher journal like Science or Nature or ProcB or something like that.
Yeah. It's I don't know. It's frustrating that that's how we have to think about. Like you're angling almost for the journal versus there can't just be like something for, good quality science almost. Like
exactly.
That being the threshold versus like, oh, this is, interesting to X population and this is interesting to this Y population. It's like, no, can't it just be like, oh yeah, we have good science and that be that?
Do you have like a almost no frills, low pressure journal for that in genetics? Because I feel like for us that might be like PLOS or the Journal of Vertebrate Paleontology, especially if you're describing a fossil. They might take a little bit longer, but like people in the field read it and it's kind of like respectable, but nothing too fancy.
I guess the best one of that is the journal G3. So G3, I'm looking at it. Lita actually got the cover for it before. It's Genomes, Genetics, and Genes is what it stands for. And that's one where you can go ahead and put in, here's a bunch of sequencing results, and here's what we think it means, but we're not going to test anything. And it's actually, they have a connection to the journal Genetics, so they're by the same group. And so if you don't get into genetics, they'll be like, do you want to consider transferring your paper to G3? Would transfer the reviews over too, and it tends to be a little bit more lax. It's not a guarantee that you'll get in, but it's much more of like we'll be more likely to let you in kind of scenario.
Gotcha. Yeah.
Cool. Shall we move to paper number 2?
Yeah, let's do it. Let me pull that up. Yeah.
Unless we have any final thoughts on this one, but I think we had a pretty good review on this paper. I think we're
I liked it. Yeah.
There we go. It's interesting to see this in communications biology as well.
Uh-huh.
Like it's a pretty good journal, and so I was like, all right, where are we going with this? This is cool.
This is thought provoking, and I really like it. So to give a little bit of background for everybody, those in chat who are not familiar with these plants, this is a cycad right here. It might look like a fern. This one's actually plastic. If I had real ones in the house, the cats would be chewing on it, and they're toxic, so I would not recommend that. But yeah, there we go, cycads. So these are a group of plants that existed before the dinosaurs and during the time of the dinosaurs and still exist today. But this is like kind of, in popular culture, almost your quintessential dinosaur plant. So like, if you're watching a movie where somebody travels back to the age of the dinosaurs, chances are they'll have a bunch of these, usually Cycas revoluta in particular, the sago cycad. They'll have those all over the place, and they're just like kind of planted in weird spots where they wouldn't normally grow. They threw a bunch of these in Walking with Dinosaurs in order to make like a redwood forest look more like a Jurassic redwood forest. They just kind of scatter cycads around, and it works. It's like kind of a visual shorthand for being like, yep, we're in the age of dinosaurs.
I didn't realize that they did that, but did you ever?
Yeah.
Have you ever been to Disney, Danny?
In Florida?
Yeah.
Dinoland? Yeah, they've got a ton of beautiful cycads there. Yeah, absolutely.
Yeah. There's that.
Yep.
It's based on that movie, that dinosaur movie that was like Land Before Time, but animated. And I remember that whole area. They've got a ton of these plants, and even in the ride
so
too.
Many, and they're beautiful, and they're really, really well taken care of. There's some very happy cycads. There's only a few species of these around in the world today. They're not very common. We only have one species in all of North America, and it only exists I think in Florida. It's called the Kunti cycad or something like that. Yeah. Contie, I think, if I remember correctly. Should be like Zamria integrifolia or something like that.
Mm-hmm. Here we go. COO.
Yeah.
Yeah, so my parents really liked these, and they transplanted one from Florida. It was on the side of the road, and as a kid, we got out. My dad.
Wait, this one in particular?
This kind, yeah.
Very cool.
And he dug one out, and so I think it just died. Actually, there was a big freeze in Georgia where where they live. There's usually not, and I think it just couldn't handle the freeze. And they'd be like, "We've had this for 20 years." Like,
yeah.
But yeah,
another name for it is Florida arrowroot, even though they are not related to arrowroot plants at all. And if you search sago palm, SAGO palm, you'll get to the most common kind of cycad in cultivation. You see these all over the place. You see them in shopping malls, see them outside people's houses. They're popular because they're like a nice, slow-growing ornamental plant that can withstand a lot of different temperatures. So they're native to Japan, and they're one of the very few non-flowering plants that people eat. These are toxic, so like there's like this long, complex process that people on this one Japanese island use to detoxify them to make them edible. But
is that why you don't have?
Really wild.
Is that why you don't have one in the house because of the cats would chew on them? Is it like toxic to the? Okay.
Oh yeah. They chew on this one all the time. But
That's
it's just plastic. So.
Gotcha.
Yeah, I do have some real cycads here, but they're in the backyard where the cats can get at them. So yeah, I've got a little collection. And for anybody who like falls in love with these plants over the course of this stream, there's a wonderful book that you could look into called Cycads, and it's just called Cycads by I don't know. I'll find it in a little bit. There it is. Yeah, by David L. Jones.
It's kind of the authoritative work on cycads, full of beautiful pictures. Every known species is featured in here, and so yeah, that I think is it right there. Yes, indeed.
Nice.
Yeah. These are really, really popular plants among plant people because they're unique. So they might look like ferns or they might look like pine trees, sort of, but they're neither. It's thought their closest living relative is probably the ginkgo. And there's something like maybe 200 species of cycad around today and like 7 genera. They are not common in the northern hemisphere. They're a little bit more common in the southern hemisphere, places like South Africa and Australia. But they used to be dominant. There used to be thousands upon thousands of species of these all over the world. They used to be like some of the most common plants around. And then you get to the early Cretaceous period, and it seems like their numbers start to decline a little bit as the angiosperms just explode in diversity. The flowering plants start to take over. But yeah, because they were really common back then, and because people look at modern cycads and they go, oh, well, they look pretty similar to the ones from the Age of Dinosaurs, they've gotten this label. People started informally referring to them as living fossils. And it's a goofy kind of a term because, well, obviously you can't be a fossil if you're alive. So you gotta be dead to be a fossil.
What do you think of that term in general? Like, not just applied to this scenario, because the coelacanth. The coelacanth is sometimes described as a living fossil, or
which it's not, which is really funny. Yeah. So it's a term that people love to use because it's evocative. Like, oh, living fossil. It's like a quandary. You know, there's a certain irony in it that people just go, oh, that sounds really cool. But coelacanths are actually very different. Our modern coelacanths, like Latimeria chelumnae, the West Indian coelacanth, not West Indian, sorry, the one off the coast of like South Africa. Anyway, it's actually pretty different from the ones from the Age of Dinosaurs in a lot of ways. People talk about, oh, this is fish that has remained unchanged for 70 million years. That's not true. They've changed a lot. To me, when I hear somebody use the term living fossil, referring to a plant or a fish or whatever,
Raiders, welcome the heck in. My BC, how was your stream? Y'all were playing Don't Starve together. I hope y'all did not starve and that you survived. Oh my goodness, that sounds terrifying. Raiders, welcome on in. My name is Valent, 1/3 of Science Streams. The other 2/3 are my lovely wife, Lita, and our daughter, baby, Alona. Lita and I are research scientists with our PhDs in molecular biology from Dartmouth, and we do live science content here on Twitch 7 days a week. Today we're joined by Professor Danny Andusa of Paleontologizing, single greatest SciCommer on the planet. As he nods, it's fine. It froze when you tried nodding. The Twitch knows that.
When I tried shaking my head, yeah.
And we're
not nodding in agreement.
Exactly. No one will ever know, Danny. It froze. We're talking fossil science, genetics, and more. Y'all, welcome in. I hope you had a great stream. Let us know how it went. We'd love to hear about it.
Hey, maybe. See how you doing? Welcome.
And yeah, we're just chatting about this idea of a living fossil. This paper looking at cycads was arguing that this isn't a living fossil. Turgus! He tried to get me. He didn't get me. The redeem is on a randomizer. Either we have a nice little roller coaster ride, or I get dropped down, just like from the movie Emperor's New Groove. Playing Don't Starve together, and somehow cordyceps ants came up. Ooh, very nice, Mai. Well, you're in the perfect place. We don't love cordyceps, but I do love talking about the cordyceps fungus and how it affects behavior, because it's having an external parasite affect the behavior of an animal by modifying its gene. Like, it's wild to me, and we know so little about it. So Yeah, that's awesome. Well, welcome in. And some animals have a mouth and butt. They do. In fact, we've looked at them
Yeah.
on stream, like the planarians. We've actually watched them eat and poop out of the same orifice live on stream because that's just what we do here. Very cool. Welcome on in, y'all. Thank you for the raid. And Kennedy, hello. Welcome in. I have a paleontologizing tea that just arrived, and now I'm the coolest kid in my neighborhood. Kennedy, may I also suggest the mug that Professor Anduza was holding, also for a paleontologizing official brand? No, that was... Sir! He knows very... There.
this one here. Yeah.
I love my blue paleo
fresh
shirt.
today. Yeah.
It's my blue paleontologizing shirt. I think I need to get a new one because I wear it so much that it's just been like etched on. But your logo, Danny, just as an aside, like you made that logo and it's so freaking cool. Like it's such a
I made that
well
years
done.
ago. That was back like 2016, 2017. I made that.
That was for like the
back when I
YouTube
still lived in
days,
Montana.
right?
Yeah.
Yeah. No, it is a really, really cool. Like it is so freaking like.
I'm glad you like it.
Way back when we first met you too, like Lita and I were like, that is like the best logo ever because there's dinosaurs coming out and it's like the accurate ones too, like the morphology and like the logo, and then it even has like the little like the instrumentation hidden away in the corners too, and then underneath it's like, it says with Danny and
It's a little busy, but
No, sir, that's busy. Okay, all the bubbles and all the characters. It's just like the aesthetic, because you have the triangular aesthetic of it, and it's just like, all right, this guy is professional. He knows. Yeah, Claire's just like, Claire's like, I got y'all. But so we were. Oh yeah, and Danny needs to collaborate with a measuring jug manufacturer. Oh yeah, no, absolutely. Those real men grow beards.
I literally got this at the dollar store like 4 years ago. I don't know if they could offer me very much in sponsorship deals, but yeah.
I mean, they just sell like what the brand did, so it's dishwasher safe. You too could be a paleontologist. Yeah. Oh yeah.
I like this idea. Yeah.
So y'all, we were talking about the idea of living fossil. In this paper, they're trying to dispel the idea that this plant is a living fossil. But Danny, you were highlighting to us why you felt, like for example, some animals labeled as these living fossils are not actually what you think, because like the coelacanth has evolved. It's not like in a period of stasis where it's just holding during time, right? And I feel like that concept kind of applies to this paper as to why it's a misnomer.
It totally does.
Okay.
It totally does. So, when someone says that a certain organism is a living fossil, I think what they're usually trying to communicate, or what they're talking about, is that it's a critter that might look kind of similar to other critters that existed tens of millions of years ago, that today are very rare. So that's the thing, is that it's like, it'll be almost like a holdout, or like a member of a group where it's not very speciose. There might be just one or two species left or something like that. And it's very different from its closest relatives. So it's almost like, if you use the term relic species, I feel like that would be much more appropriate than living fossil. But it still kind of implies that it's like a holdover from another time. And it's like, no, these creatures are definitely adapted to today's world. It's just that they're almost extinct. So like a tuatara would be a good example, where there's this creature that looks superficially like a lizard, but yet it's, from an extinct group called an otherwise extinct group called the rhynchocephalians. They used to be really, really common, but tuataras today they're a living fossil. Well, that's because they're just kind of like the last of their kind. And when you actually look at the details of their anatomy, they are so different from the ones that lived back then. But in the grand scheme of things, they might look like, they might appear to be similar, just because there's not a lot of in-betweens anymore. It's like we've got one representative today, and it looks vaguely like the ones that lived tens of millions of years ago, so we're going to call it a living fossil.
because this is a once wide-ranging group that is now almost extinct.
So is that why they might also call the ginkgo a living fossil?
Yes,
And
absolutely.
and the
yeah.
same
Yeah.
reason would apply as to why it's not, right? Because it is
Yep.
in fact adapted to live in today's world, which actually, like we talked about with the
Absolutely.
ant paper, right? There were so many variables that changed in addition to the rise of angiosperms that could have affected
Yep.
diversification. So presumably if you picked up a plant from before those changes happened back in time and you plopped it in our world today, it would probably just die, right?
Probably, yeah.
Because
Yeah.
like
I mean, with
rough-
modern diseases, with the whatever else, yeah.
Yeah, oh yeah, diseases.
Probably the nitrogen fixing bacteria that they're symbiotic with, or whatever else. Like, yeah, they'd be toast.
Yeah, so it's okay. It's a really cool, I think, graspable way to think about it as well as to why this is
Yeah.
not an appropriate way.
And for cycads in particular, like for some of these other groups that I mentioned, you mentioned ginkgos, and there's one species of ginkgo left when there used to be thousands and thousands and thousands. We have got, I think it's like 10 species of ginkgo just from the late Jurassic Morrison formation, when Stegosaurus and Brontosaurus and Allosaurus were walking around. There's like probably at least 10 species of ginkgo in the same environment, something like that. Today there's only one in the entire world. Cycads are not like that. There's like, what, 200 species of cycad? Let me look it up. But they look pretty different from one another, and they look pretty different from a lot of fossil cycads.
They have
They're
375 species in 2 families.
There you go. Oh, yeah.
So
and there's,
huge.
hang on, there's 7 genera, I think? Don't tell me. I'm going to try and rattle them off the top of my head. There's Zamia, Ceratozamia, Lepidozamia, Encephalartos, Cycas, and Diune. I think, oh, that's 6. I'm missing 1. Shoot. But yeah.
So
What's the other one?
So it's Boania, Ceratozomia, Straduria, Zamia,
Yeah.
Encephalartos,
Okay.
like you said, Macrozamia, Diune, and the Cycas. I'm probably butchering those pronunciations.
Oh, some of mine were out of date. Cycas, Diune, Macrozamia. I guess maybe Lepidozamia is now Macrozamia or something.
Yeah, they said that there's now
Nigeria.
9 genera. So there must have been like a
okay,
split
okay.
that they did.
Yeah. Shoot, maybe my book is out of date. The one that I recommended.
Well, I think it's
this
hard,
is
right?
lovely.
Because with
I love this
species,
figure, by the way.
they're
Huh?
constantly reclassifying, resequence. Like there's just you buy a book today, by tomorrow it's probably going to be outdated.
That's why, yeah, I don't know. When people ask me, like, what's the best dinosaur book, to buy to be up to date on dinosaurs? Like, well, dinosaur science moves so fast. Just Wikipedia. Yeah. I don't think there will ever be another really, really good, comprehensive book on dinosaurs ever made, because the science just moves too fast, and by the time the book goes to the printers, it's already out of date. So, yeah. What do you think of this figure here, Bullet? Because I kind of love this one. This is I've been seeing more and more of these in different paleontological publications, and even like modern zoology publications, and I love it. But I know how to read it, and I'm not sure everybody else does.
I was gonna say the same thing. I love it. I think this is
Nice,
amazing.
nice.
This figure alone convinced me of the finding.
Nice.
Because it's taking
That's a good figure, right?
Yeah, right? Like
Yeah.
it gives you, if you're already wanting to talk about diversity of the plants, right, and how they have changed and how they are looking different. Like first of all, having images, to me, I'm very like visually oriented, so they have images of representative species, and then you have the evolutionary time, and then the diversity just with the tree of life here.
Yep.
As I'm like, this is great. This is very straightforward. I get this. I'm on board.
Yep. I really like it, and I. Do you recognize the different colors that you see in the little time chart there? Those are like the standard colors that we use for different time periods. So yellow right there on the far left, that is the Neogene. Orange is the Paleogene. In green, from 66 million years to 145 million years, that's Cretaceous. Jurassic is the blue there, 145 to 201. You have Triassic in the purple, and then you've got Permian, and then you've got Carboniferous before that. And those are like the standard colors that we use. You can find them on geological maps and stuff. You can find them on cladograms or on time charts or whatever. So that's kind of like the world standard for those different time periods.
I'm jumping over to the ant paper real quick because I'm just curious if that was also
It's
maintained. Is it?
Yeah,
correct?
the light green is Cretaceous.
Okay.
Yeah. And then
Oh yeah,
you
look,
should
there's
have
the yellow
Paleogene
too. Okay.
in orange, yeah.
Okay, cool. That's really I don't know. I was just for some reason I was not expecting it to be correct for some reason. But that's really cool. Okay, nice. Heck yeah. I like that it's standardized. That's nice to be able to look at any one of these figures across the field, right, and to take into what the meaning is like a little bit more readily, I think.
And you can look at a geological map, like just search like geological map of, I don't know, what state are you in right now, Bullen?
New Jersey.
New Jersey. And as long as it's not one that has like tectonics, as long as it's like the bedrock geology, then those different, there you go. Yeah.
Oh wow, yeah,
so
look.
those are going to correspond to different times. See?
Oh, nice.
There you go.
That's really cool.
Isn't that cool? Yeah.
Huh.
So isn't that neat, chat? The kind of standardization like this, I think it's the ICS, the International Council on Stratigraphy, kind of sets the standards for colors and for dates and for everything else. So that way we don't get too confused. You know, you've got consistency like that. Not everybody's using a different color scheme and all that. So, yeah, I don't know if they have it optimized for people who are colorblind, but it works for me. I don't know.
And what's actually really cool is, so where we go hunt for those fossils is right around here, and you can see it's
Cretaceous.
in the Cretaceous, which is again, that's really, really cool. That's super cool to be able to see, and it's like further solidifies, like, again, like we knew what we were already looking for, but it's cool. Andrea, this map, so you can Google see the exact search terms that I put in. Geological map of New Jersey, or whatever location that you're in, and then this will give you the geologic time, right? If I'm interpreting correctly, Danny, across the map of what you'd be like finding and looking.
What age the bedrock is in different spots on the map. The different color splotches are the different ages. So like green is Cretaceous. Kind of where is Haddonfield, New Jersey, anywhere near there, Berlin? Because that's where Hadrosaurus was from.
Haddonfield, let's see, would be. It's in this Camden County.
Oh, okay.
So it'd be around here. I can't remember off. Is it Cretaceous?
Yeah, Hadrosaurus is Cretaceous. It's like a marine or lagoonal marl.
Okay, so my bet then, because we're not too far from Haddonfield, actually, like physically right now, and so without doxing myself, I bet then it's around here, and this is like the Connecticut River here, flowing through as well.
Okay. If you ever get a chance to go to Haddonfield, they've got a big bronze Hadrosaurus statue in town.
Okay, hold on now. I gotta Google this and make sure I this way I'll save it, right? Let's see, Haddonfield, and what kind of Hadrosaur?
Hadrosaurus, yeah.
Cool.
Yeah, there it is, right there, top left.
Oh wow.
They dress it up for Christmas, as I understand. They put like a wreath around its neck and a scarf and stuff. That's where one of our first dinosaurs ever named from North America, right there in Haddonfield.
Again, so without doxing myself.
Yeah.
I'm gonna take a look. I probably do mean Delaware River. Will, I apologize. My geography is not the strongest. Um, it is
It's better than mine. Yeah.
Danny. It is 8 minutes from me.
Holy cow! Wow, that's awesome. You should go see it sometime. I'm
That
jealous.
is so cool. I had no idea. All right, well,
Yeah,
yeah, it's an 8-minute drive. Why not go?
that's awesome. Yeah.
All right, sir. Well, I'm gonna take the Munchkin and take her picture with it and send it on over to
Very
you.
cool. Very cool. I can't wait to see. Yeah.
Awesome. Yo, I apologize. We totally rabbit holed. I was like, Haddonfield is really close to us. Like, this must be like, it really is really close. Okay, apologies. We've totally like.
No worries. Yeah.
I got excited about that.
Uh-huh.
So yeah, with this paper, we were talking about how cool this figure was, and again, like the color standardization part is awesome. That there's something like that in the field.
That's typical. Yeah. Yeah. That's not like a feature of this paper. That's for the chat. That's just like that's a standard that we have, which is neat.
Yeah.
So I'm glad you're so excited about that.
Well, I mean, it's great because that means I can. I mean, I don't. Again, like you said, I'm not sure how colorblind friendly it is, but let's say that if I'm not colorblind, I can look at any paper and look at the colors and just get the information really quickly. Because I mean, it's nice that they have like the numeric delineation here, but just to be able to go in and immediately capture that information is really, really powerful.
Yeah. That's so funny. That's Socks sounds like a soundboard. It's such a perfect meow sound in the microphone.
Jeez.
Or is that Phil or a different cat?
No, this is Socks. No, this is Socks. She's
Oh yeah.
she's our because she was in Lita's mom's barn without a home, but then she was really sickly, so we brought her inside, and like she immediately knew about the couch to get into, like sit on a couch, and like wasn't scared of the TV and like loved Snuggles, and so it was like there's no way she was always a stray, right? Because she was like so friendly. And then Lita's mom wasn't able to take care of her anymore, and of course I was the one. Whenever I went to her place, for like 45 minutes would go by, and I'm like, "Where's Balint?" Oh, he's probably petting Socks. And then so when it's like, "Well, she needs a home," I'm like, "God, all right, well, so and she knows that I'm the sucker because she's like always in my lap snuggling and meowing at me. I'm like, well, this is why I can't go to pet shops or anywhere nice, because I'll just bring home animals too.
Yeah, I can relate. Yeah, there's a similar story for basically all three of the cats who live here too. So, yeah.
Well, and also like we know Ios and Lordy are so warm-hearted that I can't imagine either of them would be anything but like, yeah, come on, like.
Yeah.
I mean, that's one of the reasons too, y'all. We've shouted out Ios and now shouting out Lordy too, of like one of my very first follows on Twitch, because you could tell like there was warmth in them just on the screen. I was like, all right, well that's already, you can see it's a good person. And then of course cats started showing up, and there's the cat cam for Ios like on the starting soon screen. I was like, all right, we're done. These are good people right now.
A big testament to their warm-heartedness is now I live here too. They adopted me as well. So, pretty grateful for that. Yeah. And Sweetie Pie's here in the room. Maybe I can entice her over with a treat. Ooh. Yeah, come here, Sweetie Pie. We'll see. But yeah. Anyway, not a whole lot else to discuss with this paper, I guess. The figure right there is just, it really captures it in a beautiful way, I think. And you can see just the diversity of these different leaf structures and these different cycads today is pretty impressive. Like, this is not, the last relic of an ancient lineage. They're really diverse today in terms of their morphology. Like, huge disparity in the and it's just how they look. And there's got to be huge genetic disparity too in that.
I always like to think of as to why we have this paper, right? And so it's one of those things that seems like with this figure very straightforward, but why do you think in the field this was something that was even needed? and it's in a good journal too. Like this is a pretty impactful journal too. It's
Yeah.
I'm just curious as to why we thought like why it's being so largely received, and it's I guess it's like how you know how you said with the rat, like of course, like I don't get it, that is his imagination, yeah, of course, but like
Yeah, I see what you're getting at. Yeah, and I think the authors kind of bring that up toward the beginning of the paper, where they say that this term "living fossil," despite not being a good descriptor for this group, it's something that gets talked about in the popular press, and then it gets talked about even in scientific journals, and it even has an impact on funding, where it's like if you can work that phrase in "living fossil" into a grant proposal or something, that's going to get people's attention. And I think they're saying that the way that it colors our thinking about cycads is not helpful, because this actually is a really diverse group, morphologically very diverse. Disparate, maybe is a better term than diverse, because they're not super speciose nowadays. There's what they say? They say 375 species. Is that what it was?
I think so.
That's
Yep,
way more than I
375
thought.
into 2 families.
Yeah. I think it's just trying to correct some unproductive ways of thinking about cycads.
It is
I
it
don't
is
know.
really interesting that what you just said too for the grant perspective. I did not realize that that's a buzzword. That
It
is
is. Yeah, well, that's a perfect way to put it, is that living fossil is very much a buzzword.
Mhm.
Loosely defined, it doesn't really, it's not a super useful term, but it's something that gets people's attention and gets them excited. And I think as scientists, it's important to examine anytime we have a term like that and ask, what is it really doing? You know? And is it muddying our thinking at all? Is it clouding the way that we think about this group of organisms in a way that's not helpful? Yeah.
yeah. It's a really I'm stuck on that, Danny. And I shouldn't be. I shouldn't be, because there's the same thing in genetics too, right? There's definitely these buzz terms that are, if you funnel it like hot terms, if you put in phase separation right now, it is like a hot word to like funnel into a grant, and you'll automatically get some eyebrow raised. But it's really interesting to be like, well, this phrase, like phase separation, we know is like a thing that happens, but this term living fossil is so fundamentally wrong that you're awarding a scientific merit on the wrong principle.
At the end of the first paragraph, it says, surprisingly, though, this metaphor still holds substantial power in swaying research programs and generating hypotheses.
I guess when I read
so
that,
you
I
think
did.
better than this
I
as a
didn't
scientist.
realize.
Yeah.
Like I just kind of was like,
Yeah.
you know, like it's a buzz term, cool. But I just like the fundamental effect of it, that it's for funding that it pulls in. Like, I guess this is disappointing. I guess.
A little bit, yeah. And presumably these authors publish on cycads a lot and are very passionate about them and are, they're eager to change this misperception. And good on them for it. This is cool stuff. Like, I now have a go-to paper that I can return to on stream when people ask about living fossils. Talking about this as a non-helpful.
Yeah. Ask a Scientist Gaming, welcome on in. Raiders, how was your stream? Were y'all chatting with scientists and gaming, perhaps? Welcome the heck in, Raiders. My name is Balint, 1/3 of Science Streams. The other 2/3 are my lovely wife, Lita, and our daughter, baby Alona. Lita and I we're research scientists, our PhDs in molecular biology, and we do live science content here on Twitch 7 days a week. Today's special guest, as always on Wednesdays, Professor Danny Anduza. Name in the bottom of the chat today, or in the channel episode description. Single greatest psychomer on the planet. Welcome the heck in, y'all. Ask a Scientist says tonight's guest was Dr. Roberto Vincis, neurobiology of taste, reward flavor processing, real-time monitoring of neural activity, and Sonic 1. Oh, very cool. And for those of y'all who don't know, Sonic the Hedgehog is the namesake of developmental genes in biology. The first one discovered in fruit flies is called Sonic Hedgehog because the scientists A, liked the video game, and B, the embryo had these little spikes coming off it that looked like Sonic the Hedgehog.
Wait, but where did Sonic come from? Because Hedgehog came from that. Was Sonic just a term that was being used for other stuff before, like Sonic this, Sonic that?
Oh, no, that's the video game character is Sonic Hedgehog.
Oh, no, I know Sonic the Hedgehog. I know I don't do a lot of games, but I've heard of Sonic the Hedgehog. Yeah, goodness. There's an echidna in there, too, right? Not a non-placental mammal, not a female. But so they named both parts at once, Sonic and Hedgehog, because there's also Sonic this and Sonic that.
So the name of the one gene is Sonic Hedgehog.
Uh-huh. Yeah.
And then that's one discovered in flies, and then there's two more that are in mammals. So there's India hedgehog and desert hedgehog.
Oh, it's the hedgehog one that's the more prevalent.
Yeah.
Okay, I got you.
That's sorry.
Yeah.
I know you play video games, Eddie. I'm sorry. Well, there was was it this week or last week? You're reviewing it was last week, the Jurassic World update with the fluorescent dinosaurs. Yeah. That's.
Yeah.
I just because in your trailer, right? It's like if you're looking for video games, you might be in the wrong spot. I just thought, it resonates. It resonates.
Yeah. No, of course. I don't know anything about video games. Yeah. But at least I've heard of Sonic the Hedgehog. Yeah.
But yeah, Raiders, welcome. We're chatting fossil science tonight. We also were looking at the evolution of ants in relation to the explosion of angiosperm. So it's been like a plant-heavy ancient science tonight. It was fun tonight too. It's fun every night, but also we had no prior really chatting about the topics, and it just so happened we were both focusing on ancient plants, which I thought was really cool.
Yeah, nice little confluence there.
Yeah, and Misadventures in Astro, hello, welcome in. All right, so I guess one question that was in the chat earlier, Danny, that I also kind of been ruminating on to ask you, is that you had mentioned that there is toxicity in these plants today.
Oh yeah.
Is there evidence of dinosaurs eating these plants, and how did they overcome the toxicity?
So there are species of animal today that will eat these. Lots of beetles, scale bugs and mealybugs really like these. And there are, I don't know if there are many vertebrates that are adapted for eating them. I know in Australia, cattle that were introduced there would get this thing that Australian ranchers called the Zamia staggers. When the cattle would eat plants from Zamiaceae, cycads, there's weird, almost neurotoxic effects, and they stumble around and foam at the mouth and stuff. But during the age of dinosaurs, it's pretty much a cinch that there would have been certain groups of dinosaurs that were adapted to eat these. We just don't have hard evidence of which ones or when or anything like that. We can speculate, and there's actually a lovely piece of art in the book here. Let me show you. Stegosaurus has got this nice bit of throat armor on it that we've only really realized within the last 20 years. And in this book called In the Presence of Dinosaurs, it's kind of like Walking with Dinosaurs in book form.
I have it. It's right over there.
Yeah, shoot. Excellent book. I'm so glad you like this one, Bullet. It's one of my favorites.
Purchased because of you, sir.
Illustration. Oh, yeah?
Oh, yeah, you recommended it. I remember we were driving home on the interstate, and I'm like, Alito was driving, I'm like, all right, we got to buy this book.
I'm glad hopefully it wasn't too expensive.
No, because Claire always links the second hand. Yeah, exactly. I'm like, all right, it was $6. I'm like, oh yeah, absolutely, bring it on.
Yeah. So in this, Stegosaurus is reconstructed speculatively as being an animal that loves to eat these fleshy fruits from the inside of a cycad cone.
And their throat armor kind of protects them from being pricked by all of these, sharp
Oh.
spines that the leaves have. So it's kind of like a nifty little explanation for like, oh yeah, maybe that's why Stegosaurus had that throat armor. Like, it doesn't look like that would protect it from a dinosaur like Allosaurus, who was eating Stegosaurus, but maybe it helped them get their favorite food or something. Because cycads are so spiny, like their leaves can be really pokey. And I've always just really loved that idea. It's speculative, but like makes sense. It's fun.
It's nice to have that
Um,
form following function.
Exactly.
Yeah,
Yeah.
that's
yeah.
really cool.
So there probably would have been species of dinosaur that ate the fruits from cycads, maybe the leaves too, and maybe even the trunks. Well, it's called the caudex, I think, is what the technical term for it. But just because cycads were so common back then, somebody's got to be eating them, and they do produce these fruits that insects feed on. Fruits with seeds inside. Um, so, yeah, but we don't really have any hard evidence of dinosaurs eating them.
I guess it
Difficult hypothesis to test.
it ties a little bit back to the ant paper in terms of that specialization, right? Like you said, there's so many of them, someone's got to be eating them, so there's going to be a niche that evolves.
Yeah.
And so I guess that's another reason why it's important to note that there are a lot of these species, right? It's not that they just totally went extinct like the hell ants prey, but they stuck around.
Yeah.
And so like
Uh-huh.
that's a nice like kind of tie-in to the other paper too.
Yeah. And I should also add that with these things, so they do have pollen. Like still have to be able to reproduce via pollination,
Mm-hmm.
even though they're not flowering plants. It's thought that they were pollinated by beetles, though. And there are a lot of species of beetle today that, like, they're not well studied, but there's a lot of speculation that, like, oh yeah, this particular species of beetle hangs out around cycads a lot. They might have, like, a mutualistic relationship there. And yeah, to my knowledge, there's not been a lot of study of that kind of thing, but there should be. Cycads are really cool, very charismatic plants, and yeah, we need more people working on them.
Yeah, this is the perfect paper, like you said, to pull out whenever a living fossil comes up.
Yep. And I think that living fossil thing too also has, like, in the general public, for people who know about cycads, that's usually like the first thing they think of is like, oh, living fossil. There was an article in the New York Times like 20 years ago or something, and it was talking about this community of plant collectors, like these oftentimes wealthy, obsessive collectors of plants who were really, really into cycads to the point where, like, they were paying people to smuggle them out of certain countries, and, like, they were going and poaching them from the wild, and there was, like, a big FBI sting, like, the Federal Bureau of Investigations got involved and, like, tracked down some of these people. And let me see if I can find it, because it's a really interesting article. But cycads, while the general public might not be totally aware of them, people who know plants get really excited about cycads because there's this idea of, like, great antiquity and rarity, and they're just really different from all other plants today. So if you're a plant person, you might get very excited about cycads.
The smuggling element is very fascinating. Danny.
Yeah. Let me see if I can find this. The Cult of the Cycads from the New York Times, from 2005. Let me give you the link. It's been a long time since I've read this. I should revisit it. But
Nice.
it's good stuff. Like the seedy underbelly of the plant collecting world.
I guess it reminds me a smidge about the underbelly of the ant collecting world. Like also,
Yeah.
smuggling the ants where they shouldn't be, and the USDA coming after people.
Mm-hmm.
Yeah.
Yeah. Shoot, a bunch of the cycads from this article that were seized by the FBI are now here in Berkeley, California, in the Berkeley Herbarium, I think, or not the herbarium, but the botanical gardens.
I've not had a chance to visit yet, but I've heard really good things, and
Ooh.
I might be able to see some of these same individual cycads who were rescued during this operation.
Perhaps an IRL stream?
That would be really cool someday, yeah.
Especially like linking back to this paper, I think that'd be a blast to kind of show the variants, and also just to tell the story about the smuggling.
Yeah, absolutely. Yeah, it's cool stuff.
Yeah.
Like, what a neat group of organisms. These plants are so cool.
Very.
Yeah.
Danny, it is near time
Yeah.
for you to skedaddle and have dinner.
I can actually stay on a little bit longer.
Okay. I wanted to make sure
Yeah.
that I wanted to plug that Amazon list and the events that you're going to be doing. Do you
Yeah.
Can we do that now? I just want to make sure we have time that folks are aware, and I'm gonna.
Yeah.
Okay, awesome. I have the link ready to go, and just to show off to everybody, if any way that we can help Danny, y'all, that's something we're always
I
doing
really
here.
appreciate that, Bullet.
Of course.
Yeah. Here, I can do a little spiel while you're typing. For anybody who's not aware, I go out and do field work during the summers. So this summer I'm going to be going out doing work in Wyoming at the end of May through the end of June. So I'm going to be out there for over a month digging up at least 2 new species of dinosaur out there. And this is in an area that's like pretty much unexplored for dinosaurs. Not unexplored, but it's not really been collected in for about the past 100 years. Or 90 years, I should say. And there's 0 dinosaurs that have been named from this particular time and place. So anything that we find, chances are it's going to be new. Whether it's a dinosaur, whether it's a plant, whether it's a fossil fish or something like that. It's exciting stuff, and we've got some funding. We're not doing a huge fundraiser stream like we did last year. Last year we were able to actually fund our fieldwork primarily through the paleontologizing community, which was incredible. We have some people funding our work this year, which is awesome. People are understanding the importance and they're really excited about it. But we still need some gear. And so that's what this wish list is for. Everything that you see on here, chat, are different items that we will use in the field. Whether it's pencils or rock hammers or shovels or, coolers for food. Each one of these would be tremendously helpful to us. And so far, the paleontologizing community has really stepped up, and Ethan, who's the expedition leader, a friend of mine, he texts me almost every morning. He's like, "Yep, this showed up today. Yesterday it was I think the cooking tent and the supply tent were both purchased.
Wow.
Today, this morning it was the plumb bob, which is it's like a surveying tool that we need for mapping the fossil quarries. So yeah, it's been amazing, just the level of support. And yeah. If any of you would like to provide direct material support to a crew of paleontologists and volunteers digging up dinosaurs, here's a way to do that. So, yeah.
It's remarkable that, it's sad that it has to be through Twitch, but it's also remarkable that through Twitch, like last year, it was able to be the fundraiser to do the field work, and then now it's just like for all the tool collections. And guys, it's not like it's anything super pricey either, relatively speaking, right? It's not thousands of dollars like in a molecular lab. It's like just fundamentals for like. Actually, curiosity, what is a pencil used for?
For taking notes.
Okay. I was like, oh man, what could, like.
And so that we can do our sketch maps like of the different quarries. So we got to make maps as we're digging things out, so that we've got good data on exactly how the bones are laid and everything. That way you can tell the direction of flow from the water and the sediment buried in, all kinds of stuff like that. So yeah.
Gotcha. For some reason, I was like, oh, maybe it's like the graphite to mark on the bone was like not damaging, and it's like, or that's where I was going. I was like, oh man.
Literally just writing. But yeah, and what's cool is that most of these things too are things that we can use year after year after year. A few of them are consumable, like the pencils and the burlap. We'll use that stuff up. But yeah, like the rock hammers and the picks and the chisels and stuff like that, those will probably outlast us. There will be future generations of paleontologists using those after we're dead. So it's cool to think about that, like, you're gonna help contribute to paleontological fieldwork for decades to come.
It's amazing,
Maybe into the
and
next century, who knows?
and Danny will make a custom command for this to trigger every couple of times throughout the stream, so that we can also help y'all out as best we can.
Shoot, I really appreciate that.
Of course,
You
yeah,
do
we
not
did.
have to do that. I don't want to be
No,
invasive.
no, sir.
But
This is like a scientist helping one. It's the least. You do so
I
much
really
for the
appreciate
community
that.
of science on Twitch. It's a very small. We did the same thing for when it was the fundraiser last year too, and it's a little thing that could be done. Like same thing, like y'all when we, Danny will mention. He's like, I see you're like shouting me out on like the stream here. I'm like, yeah, it's because if we can direct people over, like if we have this
Yeah.
front page carousel, right? Let's lift each other up, and it's the smallest
Yeah.
effort to lift the other person who's also part of the community and is, I mean, I tell you every time, but you've done so much for the science on the platform. Like without you, there would be no science on the platform at this stage.
Oh, Blend, that's not true. There would still be science on this platform. Holy cow! But
In an environment where it's okay to ask questions, that is not politicized, that is welcoming and exciting, I think if you weren't doing it, it would have just died off. Or it would become this very niche thing where it could be like almost insular and ivory towery versus like you've opened it up for people to be excited and love for science again. And so, I mean, just thank you.
I don't know what to say. Thank you, Blend. Holy cow! Shoot, I really hope that I can help further that this year when I'm in the field, because I don't know if I mentioned, but I'm going to be streaming live from the field, live via satellite as often as I can, and I'm going to do whatever I can to help grow the science community here on Twitch and shout you out as often as I can, Bulent. And who knows? There's rumors I might even be able to get a front page slot while I'm in the field this summer, which would be really cool. So that might be a wonderful opportunity to kind of just let people across Twitch know that there's science going on here too, and it's exciting and interesting, and they can learn a lot. And yeah,
I mean,
can I
also,
tell you something else?
I
Go
mean,
ahead, go ahead.
just that slot. Like, I
Uh-huh.
can't imagine how many people's imagination you'll recapture, who had a love of science as a kid, and seeing someone digging for dinosaurs. That can have such profound implications. I know I'm not, like it could be immense right there. So I really hope that comes through.
That dovetails perfectly with what I was just about to say. When I was a kid, when I was 10 years old, there was this program that came on TV. My dad taped it for me on VHS. It was on A&E when it used to be Arts and Entertainment Network. They used to show like operas and stuff like that. It's very highbrow back then. Not so much today. So they were like experimenting with a lot of like educational programming and stuff. And the television network funded a paleontological dig in Egypt. The program was called The Lost Dinosaurs of Egypt, trying to rediscover some of these fossils that had been destroyed in World War II. Basically trying to like rediscover Spinosaurus. And so basically it was a 2-part series narrated by Matthew McConaughey. And it followed a crew of paleontologists and geologists as they went out to Egypt and searched for dinosaurs. And I was transfixed by this as a kid. I thought it was the coolest thing in the world. Just like having a camera crew following around a crew of paleontologists digging up dinosaurs. And I'm trying to kind of recreate that in some way on Twitch. But here's the cool thing. Something I just learned recently. One of the lead paleontologists from the Lost Dinosaurs of Egypt, is going to be joining us this summer in Wyoming, it looks like. And so, growing up, watching this guy in this program and being like, oh, I'm going to do that someday. That's going to be me. And now he's going to be joining us on my show is going to be, it's a weird feeling to think about, kind of a full circle sort of thing. So we'll be watching that program before I leave for the field on my channel and just getting hyped for that. It's gonna be good.
That is mind-bogglingly cool.
I almost like don't want to talk about it because I don't want it I don't want to jinx it somehow, but
I
yeah.
I get I totally get that, but that's gonna be amazing. And just just like you said, the experience of you as a kid and kind of what you got excited, your dad recording it, and then now like it's just, I would imagine it's very surreal. It's like, for me, that would be like Jeff Corwin
Yeah.
was like the big one that I watched as a kid, of like when he went and
Me
explored.
too,
Like,
actually. We were just talking about Jeff Corwin today for World Frog Day. But
Yeah.
yeah. So yeah, I don't know. I'm trying to think hard about how I can make these streams as accessible and exciting and interesting as possible, so that we can really get people interested in fossil science, and make them feel like they're right there digging up dinosaurs with us. So, yeah, it's something I'm trying to be as thoughtful about as possible, because it's a big responsibility, in a way. Like, having people watch and spend their time and their attention on what we do it's a big privilege, and thank you for doing such a good job of making this worth people's time, making it as accessible as possible, making it entertaining and engaging, and not sacrificing the quality of the information. You do a really, really good job of that, and thank you. Because I know it's not always easy.
No, I really appreciate that, because that's always the balance, right? Of like, it's not that you're dumbing it down, it's you're making it accessible, right? But you can sacrifice it if you don't do in a correct way, or if there's not I know the prep work that you do going into one of these streams is insane, over the top, and so it's really remarkable of everything that goes into one of these. And again, thank you for everything that you do, sir. Right back at you. Guys, if you're not following Danny as he's changing the world on Twitch, here is the Fancy Pants shout out again and again. He'll be in the episode description today. Highlighted name. Please go follow him. You're almost at 20K, Danny.
For followers? Am I really? Shoot.
Did we pass it already? Hold on. Did I miss the 20K celebration stream?
I thought I was still at like 18,000. But I haven't checked in a while. It's loading.
Oh, 18.9. We're almost at 20. We're getting there.
Okay.
I get excited for it, because I'm like, that's showing like the huge like interest in the sciences as well. Yeah.
For sure. Yeah. And shoot, Balloon, we've got to talk to folks at Twitch at some point soon about another panel discussion this year at TwitchCon. Shoot.
Fingers crossed.
Let's talk about that soon.
Yes, we'll message. Before you go, Danny, Claire wants me to show you something. We had a art request by Claire to do an Antonia in the style of a paleontologist, with you being the inspiration. And so now we have this animation that is in homage of the crossover. So.
Very nice. I really appreciate that. Holy cow. That warms my heart.
So Claire spent 100,000 channel points, and she was like, make sure to get the hat.
Holy moly.
The hat, the necklace, the clothing as well. So we tried to nail that down, and we had the dino pop out, and so it's like we always promote the crossover stream, and so now we're starting to use that image a little bit to get people again like the crossover happening. And again, y'all, Danny doesn't have to do this. He does this because he's a really kind friend, and we are able to.
Because I enjoy it.
Well, I really.
And I thank you for spending time with me, and it takes time. You're streaming here 8 days a week. You work 3 jobs. You've got a wife and a kid and 19 cats.
It feels like.
It's just the fact that you find time to read papers that I send every week it means a lot.
So thank you.
I really look forward to it. Like every time it's even though it's outside my field, I learn so much from our crossovers, and I think it's nice to be able to have this interaction with the sciences and chat with each other and appreciate each other's fields, and not have it be this like insular idea of I'm this kind of scientist and which y'all does happen. It really does. And so it's really nice to have this overlap. Actually, Danny, I was telling about there is this researcher I'm going to try to get on April 8th, and he developed a way to isolate DNA with at-home tools, like soap, contact lens solution, isopropanol that you can get from CVS, and a coffee filter to isolate DNA. And so we're gonna try to do we're gonna do a live stream where we're gonna go through the process, and folks can do it at home. And so that's one of those things too. It's like breaking down these walls of like making science accessible, and that's what I really like about these crossovers too.
That's really neat. You're gonna have a supply list and everything in advance for people to be able to
do it along at home, and anyone who does it along at home and takes a picture, we'll send like a sticker to as well. And so it's just
that's super cool.
And it takes like I think I did it with him like 10-15 minutes. It's really cool. Yeah.
That's really exciting. Awesome. Well, neat. I'll see if I can tune into that. Shoot.
There'll be a Monday, and I think he's coming in around 8, but we're doing it 2 different ways. One that you can do at a bar with a shot glass, and another one that you can do in your home. So both family friendly and for, if you're just out wanting to impress your friends, you got 10 minutes.
What kind of organism are you extracting the DNA from?
Myself. So you can number one is you can do it works with spitting in the cup.
Organism.
And 2, there's fruit that have a lot of different DNA, like a lot of replicate. Yep. So like bananas and strawberries and onions.
Yeah.
So for the instructions list, it'll be like any one of those you can get, and
super cool.
Yeah,
I've heard of DNA strawberry experiments at home, so that makes a lot of sense.
Yeah. So those are the hope, and we're gonna put it out on the Discord, the instructions, and that's some of the things. But before you go, Danny, real quick, anything last thing to promote, my friend?
Let's see. Tomorrow's National Flower Day, so we're going to be talking about the rise of angiosperms on my channel tomorrow, and we'll try and cruise through Thursday Birds Day really quickly too. I'm starting extra early tomorrow because I'm going on a little trip in the afternoon. So starting like hopefully 2 or 3 hours early.
Okay, so that'll be like
and then ending early.
2 p.m. Eastern-ish?
Yeah. I'm trying to start probably like 11 a.m. California time, which would be 2 p.m. Eastern.
All right.
Yeah.
I got your schedule.
Awesome. I know, yeah. I appreciate your enthusiasm. Holy cow. And then I'm not streaming on Friday, because I'm going to be on a trip, but yeah. It's I've got exciting stuff coming up. We're looking into doing more interviews and getting ready for field work and a bunch of stuff like that, so stay tuned.
Awesome. Danny, thank you so much for your time today and for the crossovers.
Thank you. Of course. Yeah.
Please have a delicious meal. I mean, listen, it's Lordy. You're gonna have a delicious meal, but
she's making banh mi tonight. So
oh God, that's
really
gonna be so good.
Yeah.
Thank you so very much, Danny. Have a great evening. Thank you again for coming on, as always, and we'll see you tomorrow at 1 p.m. Eastern, or 2 p.m. Eastern, on Danny's channel.
Great. It's been a joy, Bullen. Thank you again.
Thank you, Danny.
Take care.
Have a good night.
You too. Bye-bye.
All right, y'all. That was the lovely Professor Danny Anduza coming on with us again. Always a highlight of my week. Thank y'all for hanging out and chatting with us about the